So, ladies and gentlemen, welcome and thank you for your attendance in this conference called Sustainable and Innovative Solutions in Agriculture and Food Industry. This biannual program starts today. This is the first one. We are planning to give you up-to-date information in the topic of One Health, so plant, animal, food and environmental health. First of all, I would like to call Professor László Csernok, Vice Rector for Scientific Affairs of University of Debrecen, to make the opening address. Thank you.
Ladies and gentlemen, dear guests, it is my pleasure to welcome you all here in Debrecen at this very nice building and very important topic. The university, you might know, has recognized this symposium as a Debrecen University Symposium. This recognition was not undeserved, as we all understand the importance how changes in environment, changes in climate, and of course changes in demographic values affect our daily life. What happens in the feed and food chain nowadays will affect the health of and sustainability of how agricultural products are made, how they are consumed, and what they will do with our health. Being a professor in the medical field, however, working also on questions with nutrition, we all recognize that the changes in climate result in altered infection of crops. If I may say just one, the warming environment in Hungary results in the spreading of previously unseen fungi and affecting aflatoxins in the food and thus feed and so on. And there are many others, of course, not talking of water problems and so on. And all these can only be tackled if experts like you here at the conference will look at these problems with appropriate scientific attention and come up with sustainable and acceptable solutions for the future. I strongly hope that this conference will be an inspiring place for you, and all what you will learn, you will have and be able to use it in your daily research. However, please note that Debrecen is also a city of beauty, so I do urge you that after having important impressions of science and nice talks and good discussions, for which a symposium is the best place to do visit Debrecen and enjoy not only the science, but also the sights that our city and university can offer. With this, I wish you a pleasant conference and a pleasant stay here in Debrecen. Thank you very much for coming.
Professor Csernok, thank you for your opening address, and especially thanks for the financial support, the significant amount of money that made us possible to organize this conference. So, we start this interesting event. I hope all of you will enjoy it. This is a kind of unique in several aspects, that in English exclusively, organized and run in English exclusively, and also to demonstrate the versatile research activities of our partners, of our fellow researchers and our colleagues. So that's why we invited speakers from all over the world, let's say, not just from Europe, but also from Australia. You will hear 9 presentations today, each and every one of them connected to One Health as it regards plant, animal, food, or environmental health. So I hope that you will get some information useful, and there is also a possibility to ask the presenters after the first and the second part of this today's program. I will call if you have any questions, and my colleagues will help you with the microphone. One other thing is that I have to mention that this conference is online streamed, so those of you who are not here can also follow this the happenings today. So we will start with the morning session. Then there will be a coffee break at 10:15, and the morning session ends 1 o'clock, and then we will walk to the university canteen for lunch. I think that that's all for technical and for organization matters. So let's start the conference, where I ask Professor Zed Rengel from University of Western Australia to give the lecture of Dynamics of Phosphorus in the Soil-Plant-Human Continuum. Professor, the floor is yours.
Jó reggelt. That's just about as much Hungarian as I know. Thank you very much to organizers for inviting me to this symposium. And I would also like to acknowledge Professor Silvia Veres, my long-term collaborator. I'm supposed to talk about phosphorus today, and you might think that this is a somewhat boring subject because lots of people know a lot about phosphorus. But I would like to start with something else. I would like to scare you first and talk about Overshoot Day. If you haven't heard of Overshoot Day, I'm going to have a website address in a minute so you can check out what exactly it is. And it is calculated based on how much resources we as humans consume and how well products are made. And of course, how many of us are on this planet is an important consideration. And on the other side of the equation, on the other side of the ledger, is how much resources this planet can provide to us. Should probably stay a little bit back. And this is how Root Overshoot Day looks like. If you go back to 70s, it was just about the whole year that we managed to make to the end of the year by using all the resources that planet Earth would provide, and that was sustainable. But as you move toward more present time, you realize that this overshoot day is coming closer and closer, which means that we use up all resources provided by the planet for 365 days much, much earlier. So we are talking about end of July, early August with 2020, a little bit of aberration, resources lasting a little bit longer because of COVID year and lots less resources being used for transport. So that's not good news that this overshoot day is coming sooner and sooner. And this particular year, 2nd of August, so we are past that. So we are now living from our future generations. We are borrowing from our future generations. We are using resources from the future, and that is obviously not sustainable. And this is how different countries fare. This is how long resources of this planet would last if everyone lived like people in this country. So Qatar is the first one where by February the 10th all resources would have been used up, and we would be borrowing from future generations for more than 11 months, almost 11 months in a year. Australia is 23rd of March, and if you're interested where Hungary is 30th of May, and for our Croatian friends, Croatia is just about there as well. So looking at Hungarian example, not even half a year. So clearly we are not living sustainably at all, and that's what we are doing to planet Earth. We are squeezing resources of planet Earth. And I will be talking about one specific resource for the rest of my talk, and it is rock phosphate. Rock phosphate is a raw material that is used for production of phosphorus fertilizers. So various phosphorus fertilizers are produced from rock phosphate, and rock phosphate is mined from the ground. So it is non-renewable. Mined from the ground means that there's going to be a point in the future when it is simply going to run out. And what are we going to do then? So there are a range of factors that influence that scarcity of phosphorus, and I'm going to spend a little bit of time on each of these 5 factors. Physical first, and it is talking about peak phosphorus. We know about peak oil, and we know that the date for peak oil changed. Initially it was in 1990s and then changed to 2010, and it is still kind of a moving target. And of course there is more than just one peak of phosphorus, depending on who made estimation, what assumptions we used in the model, and how the algorithm was set up. So you can see that there is a range of years from 2025, which is kind of now, and going into 2100. The one that is 2300 is completely unrealistic because it took into account resources that are hundreds of meters deep, and of course they are there, they can be explored, but not economically by any stretch of imagination. Even though I'll talk about economics in a minute, but just to remind you that if a loaf of bread costs 1 million forints, in this case, yes, there's lots of phosphorus resources that may last into 24th century. But society would not be able to function with a loaf of bread costing 1 million forints. And even if you go for optimistic scenarios toward the end of this century, there are young people in this audience who will live through that. And I must admit I don't envy you for it. Economics, it's all about price. When you have a resource on one side and that resource has certain price, then things can happen due to a range of factors, and the price has gone, for example, in 2007, the price has gone up by 8-fold, and recently, due to geopolitical events in terms of war in Ukraine, it has gone up by about 2.5-fold. So these are really big increases in rock phosphate, and that means that price of phosphorus fertilizers goes up, and then price of food for which these phosphorus fertilizers are used goes up as well, and then we have huge problems. In terms of managerial, this is a very complicated diagram which I don't want to go into, but just to remind you that it is ore that has been deposited in the ground over tens of millions of years ago, and it has a finite capacity. So as we extract from that reservoir that exists in the ground, we are going to deplete that reservoir eventually down to non-economic level. We are never going to deplete it down to zero, but to non-economic level. Remember, 1 million forints for a loaf of bread. And it's not just agriculture, not just food production that uses rock phosphate. There are various other industrial uses, but you can see that majority of it, so from around 16.5, so great majority of it goes into fertilizer production. And then in terms of food processing, there is about 3.5 million tons that ends up in humans. So out of 16.5, only 3.5 ends up as useful food, which means about 1/5, only about 20%. So that is not a very good use of rock phosphate or phosphate fertilizers, so very poor efficiency, which means that there is a room for improvement, which I will talk about later. And then, of course, there's a very big line that goes into inland waters, so there's lots of eutrophication, lots of losses of phosphorus from the system that cause additional problems in terms of eutrophication of waters, algal blooms, these sorts of things. So lots of negatives on this slide. Geopolitical one is interesting. I'm going to show you just one world map. And you would notice that Hungary doesn't have any rock phosphate resources. Australia also is listed as not colored because we exhausted all our rock phosphate resources. And great majority, the only significant number on this map is Morocco and Western Sahara at 70%. And then a range of countries with few percentage points, a little bit here and there. But the major resource is Morocco and Western Sahara. And there's a very important political connotation to this because if I presented this map in Morocco, they will be all over me because they don't consider that Western Sahara exists. They consider that this is the same country. And that is a potential problem for the supply of rock phosphate to the world because Western Sahara and Morocco are, strictly speaking, at war. It is not war that is currently consumed, so there's no shooting going on, but it is officially a war. And then institutional, it's interesting that even though it is a very important resource, there's basically nothing that regulates that resource. There is no rules and regulations, there is no institution specifically governing that particular very important resource. And that means that long-term food security is in jeopardy because it is left to the markets. And markets is supply and demand. And as supply is dwindling, demand is increasing, prices are going up, and that is a big problem. Markets should not be trusted to manage something that is so crucial for long-term viability of life on this planet, because without phosphorus and without a good amount of food produced, there's no life on this planet, because I say if loaf of bread costs 1 million forints, there's going to be shooting in the streets. So, what's the future? What can we do? Because it's not all that bad. There are avenues that we can explore in trying to improve our future, but this black line is showing projections, which means that from around now to around 2050, it's about doubling of amount of phosphorus that would be required, and then continuous increases even after that. But that is not going to be possible, and I will just look at supply measures first and then at demand measures. Just looking at supply measures, you see that phosphate rock is a relatively small part of that future supply, and that all other ones have word reuse, which means that we will have to be reusing various forms of organic phosphorus that exist in the system. And we are relatively bad at doing it now, so these are opportunities for the future, for young generation to sort of sink their teeth into it and try to improve that reuse or recycling of organic phosphorus, because phosphate rock, as we used to just rely on it for production of phosphorus fertilizers, these resources are dwindling and becoming more expensive. There are things that we can do also from the standpoint of demand measures, and most of them deal with efficiency. So all except the last one have efficiency, increased efficiency of use of phosphorus. So it is increased efficiency of mining, rock phosphate, increased efficiency of producing fertilizers, and then increased efficiency of using these phosphorus fertilizers. And then I will have the last slide on changing diets, which is somewhat controversial topic. So there are things that we can do, and this is elaborated a little bit more here, where you see that blue arrows represent demand intervention points, and it is mostly about minimizing or avoiding losses. So all these arrows that are pointing downward talk about losses. There are some losses in terms of non-arable land and losses to water. And the red points representing supply intervention points are mostly about reusing, about recycling. So there's lots of organic phosphorus in the system that we can use and manage much better. And this is an example from the past, but it shows what can be done in terms of improving efficiency of phosphorus fertilizer use in China. So if you start from the left, bottom left, you're looking at distant past and then changing colors toward more recent past, about a decade ago. But you see a continuous increasing trend. So more food produced and more fertilizers used. That's kind of good because there's obviously more food produced. So if fertilizers are unlimited, that would be a very good situation. In Africa, it's a similar type, except that it was relatively flat in the distant past, and then it's starting going much more in terms of not much increase in fertilizer use, but very substantial increase in food production because of better utilization, better agronomy, better utilization of phosphorus fertilizers. So in both of these examples, and we're talking about 2.5 billion people here for these two areas, in all these examples there is increased usage of fertilizer and increased food production. So increased food production is good, increased usage of fertilizer is not. And Europe is a contrasting example, because distant past you're seeing increases in fertilizer usage, increases in grain production, but then more recent past, red symbols, you are going in the opposite direction in terms of fertilizer usage, so declining fertilizer usage, but still increasing grain production. So this is the best of both worlds. So Europe is doing very well, European countries, European Union countries that is. And the US is sort of somewhere in the middle. There is not much of an increase in fertilizer usage with an increase in grain production, but there's no decline in fertilizer usage. So not as good an example as European Union, but still a relatively okay example because of increasing grain production without an increase in fertilizer usage. So clearly there are potentials for maintaining and even increasing food production without relying on more and more fertilizer applied. And that deals with lots of efficiencies that I've listed before. So more efficient use of fertilizers is certainly possible, and there are good examples in European countries in terms of how that can be achieved. And then to the last slide that deals with as I indicated earlier, a somewhat controversial topic because it is talking about changing diets. So without going into the left side first, and please note that red lettering that's flashing kg should be written as lower kg, so I just copied it from the reference. In looking at Ghana, there is a greater majority of food that is plant-based compared to animal-based, and Argentina on the other side, and Australia is actually worse than Argentina, but it hasn't been listed in the original publication, where a very small proportion of diet is based on plants and a very large proportion based on animals. Unfortunately, there's no Hungary there, but if you're looking at the world, which is sort of somewhere around 2/3 down this list, most is from animal-based food and a relatively small proportion from plant-based food. And now the crunchy bit is that for a unit of phosphorus, for 1 kilo of phosphorus, you can produce very different amounts of food. If you're looking at beef or generally animal type food toward the bottom of this list, you can produce relatively small amounts per unit of phosphorus compared to a situation where looking at plant-based food, particularly various tubers, potatoes and starchy roots. So you're looking at the difference of about 200 times. So if you translate that into human terms, for a kilo of phosphorus, you can feed 200 times more people if they are eating plant-based food compared to animal-based food. But of course, it is not an easy topic, and as a large proportion of the world is developing and changing diets, that change is actually toward more dairy and more meat. And I'm talking particularly about China because I go there frequently and I know the situation there quite well. So this is something to ponder about. Not that I'm advocating that everybody changes their dietary habits, but it is something that needs to be kept in mind as potentially a part of the solution for the future. And the future is not going to depend on rock phosphate, because rock phosphate is non-renewable, and whether it's going to run out in terms of economically feasible reserves in 5 years or 10 years or 50 years, it's beyond the point. It is going to run out relatively soon, as I say, within the lifetime of most of the audience. So it is something that needs to be kept in mind, and measures need to be taken as soon as possible to start to address that point. Because we will need to produce more and more food. Projections are about doubling the food production between now and 2050, and about 10 billion people at 2050. And that will have to be done with less reliance on mineral phosphorus fertilizers compared to what we have done in the past. And in terms of two important things, one is reuse or recycling of phosphorus from various organic sources that exist, and the other one is increased efficiency of use of phosphorus in the whole cycle of phosphorus from the mine to the plate. Recycling needs to be improved, and recycling needs to include not just kind of obvious things, crop residues and food waste, but human waste as well. And then the last bit is to keep in mind is about the fact that it takes lots more phosphorus to produce a unit of beef or unit of meat-based food compared to plant-based food. Thank you.
Thank you, Professor Rengel, for your presentation. It's always interesting to see that raw materials or basic nutrients for plants, like the basic of the pyramid, how can influence the food chain or the food production. So I hope that you could take these messages. I'm making notes for the presentations because I have to do the take-home message part at the end. So I will include that too. Thank you. Now the second presentation is about to come. May I ask Professor Judith Dobransky from University of Debrecen to give out the presentation, Plant Memory and its Exploitation Possibilities for Innovative Strategies for Growing Healthy Plants. The floor is yours.
Thank you. This topic is a very new topic in the sense that it was controversial, this topic, even a decade ago, but mainly to the development of genomics, epigenomics, and transcriptomics. By now, we already realize that plant has memory, and we have some knowledge and idea how and in what way it operates. So today I will attempt to address 4 questions. How can a plant remember? How long does plant memory last? Why is memory important for a plant? What is its adaptation and evolution importance? And finally, how can we use it in the agriculture for development of new plant breeding and cultivation strategies? So the first question, how can a plant remember? If you're thinking about the plant intelligence, or how can be intelligent or can be intelligent at all, then we have considered that they have no neural structure or nervous system. They are cellular organisms, so they have limited movement in space, so they are not able to run away from the surroundings and change their location if any stress occurs. But they need to respond to the environmental stresses in a changing environment for their survival. By now we are realized that plants are able to learn, communicate, make decisions, and modify their behavior. Learning of plants is a bit odd, but until we consider the definition of the learning, which is lasting and adaptive change in a system or its controlled subsystem as a result of interaction with the environment. Prerequisite for a learning is a precise memory, which is per definition the ability of a living organism to store, retain, and retrieve information based on acquired experience. When a stimulus hits the plant, and it is able for its perception and recognition, it will respond to that stimulus, but meantime, in the background, start and build the memory. In plants, the memory is based on different networks, cellular, biochemical, and molecular networks. As for the type of the memory, it depends how we can group it. Based on the molecular mechanism, it can be cis or trans, based on the type of the inheritance, mitotic or meiotic, and based on the period of the time, which will be important from agricultural point of view. It can be somatic memory or intergenerational memory, short or long term, intergenerational and transgenerational as well. As for transmemory, it means the concentration changes of diffusive signals in the plant cells, like accumulation of transcription factors, hormones, or other metabolites. In the case of cis memory, it means the chromatin change. That means chemical changes on the chromatin. For example, histone modification or DNA material modification. They are epigenetic modification. How it works. Epigenetic modification are mitotically or meiotically inheritable changes in gene function that cannot be explained by changes in the DNA sequence according to the definition of Rousseau et al. It can be three different types. One is the histone modification, which means the acetylation, phosphorylation, methylation, or ubiquitination of the histone proteins. Can be DNA methylation and cytosine and adenine sites, and can be RNA-mediated regulation when regulatory non-coding RNAs plays an important role. And this epigenetic modification can be reversible or irreversible and can be inheritable as well. And they participate in the development both short and long-term and heritable memory as well. So this is the base for the memory formation. In this summary picture, we can see how plant can remember from the stimulus perception and signal transduction to the building of the memory. So after the perception and signal transduction, memory formation and feedback starts. It means some molecular and biochemical changes, which leads to phenotypic adaptation, including biochemical variation, hormonal variation, and epigenetic imprinting. How long does the memory last? This is an important question. We know for a long time the intergenerational or somatic memory, which lasts lifelong or a part of the life of the individual and maintained mainly by mitosis. But more and more evidence we have that memory can be heritable, which lasts for one or more generations, and according to that can be inter or transgeneration. So the question arises, can acquired traits be inherited? Yes, it can be inherited. At its molecular basis, it are the epigenetic mechanisms which are responsible for learning, storing information, and transmitting that information across the generations. Now I will show some examples for different types of memory with different length when available from our own research, otherwise from the literature. So one example for the intergenerational or somatic memory, when memory is maintained by mitosis within a certain life stage, when we prime the winter wheat seeds. In our study, we ultrasonicated the seeds, and we saw that the seedling growth after 1 week was improved and changed. And in the molecular background, we found transcriptional changes, transcription factors, and some gene transcription changed. And regarding the DNA methylation study, it was detected that the DNA methylation decreased after the ultrasonication, so the seed ultrasonication caused hypomethylation in the seedling, and we found correlation between the mRNA transcription and DNA methylation changes. They occurred in the same biochemical metabolic pathways. As for inheritable memory, there are two main questions: the way of the reproduction, which can be clonal or by seeds. In the case of seeds, not only the methylation and histone modification, but also the special small RNAs are important because they are responsible for the government of the heritable epigenetic marks in the myocytes. And the other question, how many generations will it pass on? It can be one or more. And one very well-known phenomenon is the vernalization, or the memory of the winter, which is based also on epigenetic switch. And it's one of its key steps is the epigenetic silencing of flowering locus C gene, which encodes a repressor of flowering, and this related to histone deacetylation, histone 3 subunit methylation, and long non-coding RNAs. The epigenetic memory of vernalization is meiotically stable, as we know, during the female gamete formation. So, why is it important for a plant? It has an acclimatization importance when it serves as the adaptation to the changing environment and shows the survival and overcoming of environmental abiotic and biotic stresses, and allows faster adaptation to a changing environment. One example from one of our studies, we studied the DNA methylation of apple in mother plants from orchard, in vitro plants, which we are cultured under in vitro conditions for 16 years, and in the plants acclimatized from that in vitro culture, and we found that DNA methylation variation occurred in the different environment. The changed methylation pattern persisted through several mitotic generations during the in vitro culture. After we acclimatized the plant, the DNA methylation pattern started to be similar to the mother plant because the environment was similar. So in that case, it was reversible and always according to the environment. Memory play a very important role in plant environment and plant-plant dialogue. As we know, by emitting volatile organic compounds is very important in the communication. It enables the detection and release volatile organic compounds in neighboring plants and prepares them for the active defense against environmental stresses even before the stress affect them. One example for the memory behind this dialogue. In the case of herbivore attack in mice, a special VOS effect was detected. Perceivable plants was shown to be able to memorize the warning signals of impending attack of the herbivore via altering the DNA methylation, namely demethylation occurred in some cytosine sites in the promoter region of trypsin inhibitor gene. It was a short-term memory, lasts only for some days. And in this picture, I summarized some abiotic and biotic stresses with its epigenetic marks, which can be long-term. That means it's inherited from the parental generation to the offspring generation. It can be detective in the case of ozonic, soil, drought, high temperature, cold, and heavy metal stresses, and also in the case of biotic stress. How epigenetic response-induced phenotypic variation can be inherited through meiotic generation. This is one example when, for example, in the parental generation, a natural plant population was exposed to nutrient stress. It induced phenotypic plasticity of epigenetic origin. That means lower plants which required lower nutrient levels. If the environment was the same in the next generation, Robertson and Wolf detected that the adaptive phenotypic trait was inherited by the offspring, as were the associated epigenetic marks. If acquired traits can be persist for several generations, epiallias could form. Epiallias are the regions of the genome that differ in their epigenetic state, mainly in the DNA methylation, and are stably inherited into the subsequent generation. They occur in the nature and can also be induced experimentally. And the frequency of occurrence, as this is the most important, is 5 orders of magnitude higher than that of normal mutant allele. And they are subject to selection pressure just like in any allele. From adaptation and evolution point of view, plant memory can be based on these epiallias, and inherited epigenetic changes can become adaptive if offspring experience similar environmental cues to previous generation. If the epigenetic changes are related some genetic changes, for example, transposon insertion, then they can be stable or variably persist as epiallias. Otherwise, epigenetic changes can spontaneously return to their basic state. We know that rates of fitness can be increased by heritable genetic variations during the adaptation, but if selection acts on epigenetic variation, that means on epiallias, we can find faster adaptation of the population. Adaptive phenotypes rises before any genetics changes because they are faster and with a higher possibility. And mutation-limited population is allowed to adapt based on this heritable epigenetic variation. So adaptive epigenetic mutation allow a population to respond to environmental changes even in the absence of genetic variation and more rapidly. So epigenetic mutation have evolutionary effect and able to affect the tempo and the outcome of the adaptation. So by now we already know that plant memory exists and can be inherited. Basically, it's epigenetically regulated, and it has adaptive and evolutionary effects as well, and it has advantage in the rapidly changing environment. So our last question, how can we use that plant memory for the development of innovative breeding and cultivation strategies? So we live in a climate change condition, and climate change in frequent, sudden, and severe environmental stresses requires engineering flexible crop varieties having increased stress resilience that are prepared to be more responsive to occurring or reoccurring stresses of the environment within or across the generation. And that can be produced relatively quickly. I mean that conventional breeding, even if include new biotechnological methods which are based on the genetic mutants, require years or even 1 or more decades to develop new stress-tolerant varieties. But the solution can be to ensure this rapid breeding strategy, the using of the plant memory. First possibility is the teaching of training plants. This is the adaptive stress simulation based on the plant memory, and practically it has been doing for decades, albeit without awareness of its epigenetic background. Based on somatic memory mainly, And this is stress priming at cross resistance when previous stress exposure impact on subsequent stress or secondary stress. They enable plants to have more resilience to stress as improve their capacity to withstand future stresses. So this is the possibility for the development of cross-stress and multi-stress resilience in plants. In the case if we want to use the inheritable memory, when epigenetic state can be stably transmitted through minimum one, but rather more generation, then we can produce so-named climate-smart scopes. How? We can induce the epigenetic variation globally or site-specifically, and we can select for the required epigenomes. In that summary table, we can use that there are two sources of the epigenetic variation, the natural and the induced possibility. In the nature, we can use the spontaneous variation when the variation is in the epigenome. We can use the natural epigenetic variation from crosses with wild species, and we can use that the genetic changes which cause the differences in the epigenome. In the case of induced variation, we can induce that epigenetic variation chemically and physically. We can apply epigenome editing, and we can induce mutation in the epigenetic machinery. So this is the possibilities. And as a conclusion, short answers to questions posed in the introduction. Yes, plant memory is based on different networks, cellular, biochemical, and molecular, and basically regulated epigenetically. And the connection and regulation of different networks and levels requires still further research. Plant memory can be short and long-term, including also the inter- and transgenerational memory. Plants use plant memory to communicate with their environmental and with each other, and depending on its length, which is influenced by the relation between the plant and the environment, it can have acclimatization, adaptation, and evolutionary role. And how can we use it? We can use it for development of new plant breeding and cultivation strategies based on plant memory, which enable a faster adaptation to an environment that is changing more and more rapidly due to the climate change conditions. Thank you for your attention.
Thank you for the presentation. It's also an interesting thing to see that not just animals or humans can have a memory, but also plants. And I just remember my former studies that if there is a stress, if there is a negative impact on an animal or a human, that can cause death. But plants can adapt to these changes. And then we can also use it as a tool for this adaptability of plants, for making better plants and for making plants that can survive extreme conditions. Thank you. Okay, we continue with the plant part or the section with the next presentation. May I ask Professor Gabriel Ondrasek from University of Zagreb to give a presentation named Salinization and Metal Contamination in Agroecosystems, Implications to Food Safety and Security. Professor, the floor is yours.
Thank you very much, Professor Laszlo, dear Chairman, dear Sylvia, dear colleagues, ladies and gentlemen. Please accept my greetings and greetings from my home University of Zagreb and Faculty of Agriculture. Actually, my today's presentation is composed of two subtopics. One is salinization, salinity problems, and another is metal contamination. In my today's talk, salinity problems can be presented, actually my talk can be presented to this graphical abstract. On some subtopics I won't talk much, like detection. I will talk about implication of salinity problems, about key drivers and some solutions of this problem. Actually, salinity areas are very, very huge problem. Globally, there is almost 1,000 million hectares on every continent, in every country. And actually, this data is actually problematic, that those areas are increasing on annual rate up to 500,000 of hectares every year. Interesting, Hungary is also very prone to this problem. Approximately 13% of Hungarian soils, and even more than a third of soils on the great Hungarian plains are salt affected, as you can see on this some recent and other maps. Processes which induces salinization can be, let's say, divided on natural and anthropogenic. Natural are like evaporation, like arid climate, like salt minerals from parent rock material, or induced by human activities like irrigation or fertirrigation. These processes are, let's say, the most common in today's. What can we expect it over the time in salt-affected areas? You can see on these several pictures here some of the most common type of effects on areas affected by salinities are like topsoil crusting, crystallization on the surface, and of course some of the most extreme examples, salt crystallization and desertification. How we can define actually saline conditions in agri-ecosystems in terms of productive, let's say, rhizosphere? As you know, we can distinguish in the rhizosphere several phases, like mineral, gas, organic, and in the liquid phases, when actually concentration of dissolved salts exceed some threshold limit, we can talk about salinity problems. Depending on concentration of dissolved salt and some other parameters like pH, like sodium absorption ratio, exchangeable sodium percentage, we can distinguish saline, alkaline, and some other types of saline problems. All of those actually saline environments will induce different disturbances. Firstly, osmotic stress, ionic stress, and some other secondary stresses, all of them known as salt stress. Unfortunately, most of crops actually that we are producing in agri-ecosystems are not so salt resistant. We call them glycophytes versus to halophytes, but most of halophytes are actually belongs to native plants with no some big economical value. And depending on their sensitivity, we actually distinguish several classes of salt-tolerant crops from sensitive to tolerant. Unfortunately, as I said, most of crops that we now are growing are very, very sensitive to salinity, and actually in crop production, most of implications are negative. We can talk about, maybe for discussion, about some positive implication, but mostly of those implications will be negative, like osmotic stress, water and nutrients imbalances, shortening of vegetation period, plant growth reduction, yield reduction, and many others that you will see later on. On another subtopic, metal contamination, we can also distinguish some natural processes that induce metal contamination and some, of course, led by humans. Like from natural resources, some of the most present are rock weathering, erosion, volcanic eruptions, and from anthropogenic sources we distinguish, of course, waste management, mining activities, agriculture, and many others. All of those processes actually enable metal transfers from one source to another. And of course, we can distinguish some remediation approaches that can be applied on-site, off-site. But most important is actually that mostly by consumption of crops grown on metal contaminated soils, we actually induce different negative consequences in our bodies, as you will see later on. Metals, actually, when we talk about metal contamination, we assume actually a relatively large group of elements, which are some essential, but some are one of the most toxic. And actually, in agri-ecosystem, this problem is increasing rapidly and actually presents one of the main exposure routes for our body. Also, in EU countries, actually, this problem generates every year very huge amount of actually money is needed for remediation of this problem, and actually some costs cannot be actually covered by money, because with this problem some damage like soil ecological function has no value. Also, you will see later on in my presentation, interesting is that these two problems interact together and also can influence salinity. I think on salinity and metal contamination, and they enhance, actually support each other, but in certain metrics type. For instance, some of hotspots in my country related to lead and zinc contaminations are located on some these red areas down the Drava River, close to Hungary, close to Slovenia and Serbia, but also there are in some other spots. These actually hotspots are actually related with some mining activities upstream. Unfortunately, most of crops that we are producing for actually food have very weak ability for selectively extraction of essential elements and of course metals that can be toxic, as I said. And the most actually important parameters which control actually metal chemistry are, for instance, pH and presence of dissolved salts, inorganic and organic. For instance, in terms of cadmium, you can see here in this relatively narrow pH range that free cadmium pool dropped by 60%, whereas in the presence of humates, organic salts, dissolved organics, actually have been increased by sevenfold actually. So this picture shows that pH is very, very important together with dissolved salts in the rhizosphere. In the terms, when we talk about salinity types, we can actually distinguish different implications, either in so-called neutral salts, which actually reacts by neutral pH reaction, or by alkaline salinity types, which actually generate alkaline pH reaction. And depending on the matrix types, actually salt and metal uptake and distribution from root to fruit to seed will actually depend on this matrix type. For instance, on several slides you will see some of our results which we obtained in controlled conditions, studying two different actually stresses. One was sodium chloride salinity and another was metal contamination by cadmium, with four different crops, and you will see some of selected results. For instance, in terms of total yield, main factor salinity, sodium chloride in all crops markedly reduced total yield, whereas actually another factor, cadmium contamination, did not impact this parameter, as well as interaction among main and sub-factor. Accumulation of sodium in leaf, you can see that has been increased by several-fold in leaf tissue, and mostly, let's say, in muskmelon and in strawberry. In fruit also we notice increasement of sodium by several-fold, whereas other factor did not impact this parameter, as well as interaction was not significant. Similar situation was with chloride in leaf and in fruit, and opposite situation was with potassium. With potassium, which actually concentration was reduced by several-fold in leaf. And at the end of experiments, all of implication that I did mention, you can see on this picture, like leaf injuries and actually plant mortality in case of strawberry, which is known as one of the most salt-sensitive crop. But in terms of metal contamination, in terms of cadmium, we also noticed that actually salinity increase actually concentration of cadmium in leaves markedly, as well as cadmium as we did expect, but also an interaction among these two factors were significant. In peel and in pulp of muskmelon we did not notice any changes, but we did in strawberry and in case of cadmium contamination. In some other matrix type, like is nutrient solution, for instance in hydroponic, it is possible to notice that actually very short-term exposure to cadmium contamination and very low concentration, 2.2 micromolar of zinc and cadmium, can enable a progressive uptake and deposition of those metals in radish, either in root or in leaf or in hypocotyl. And these data actually support that quality of agri-ecosystem resources, like growing media, water resources, soils, etc., are very important in the public health risk, given that even under very low concentration and after short-term exposure to metal contamination, crops will definitely accumulate those toxic elements in their tissue that we can consume, and usually we consume. Also, in similar metrics in nutrient solution, but for long-term, for a relatively longer period of exposure, 10-day exposure, and in the presence of dissolved humic acid ligands, it is possible to notice that actually humic acid decrease accumulation and distribution of metals like cadmium, copper, zinc, and manganese, in comparison to conditions without humic acid ligands. In some other complicated, let's say, matrix type, like is nutrient solution with sodium chloride, and of course with humic acid and cadmium, it is possible that actually sodium chloride, like salt type, increases a proportion of one cadmium pool, increasing cadmium uptake and deposition in strawberry fruits. Actually, combined application of humates and sodium chloride enhanced cadmium mobility and uptake by this test crop. In another solid matrix type, like is washed river sand or quartz sand, actually we noticed that triple interaction was significant. Actually, that sodium chloride, humates, and cadmium interaction increase transfer of cadmium in leaves and fruits from the rhizosphere. In some other studies in different types of soil, actually was noticed that positive influence of other organic salts beside of humic acid, full acid on metal phytoextraction, and of course in different crops. On this graphical abstract, you can see actually one interesting thing. This is actually can be related to Zed's presentation, because this matrix is very, very interesting. Very complex biomass fly ash, which actually can be used as a source of phosphorus, because biomass fly ash can contain 5 to over 10% of phosphorus, and can be something that Zed talked us about reusing of some waste materials, which actually biomass fly ash is, like waste materials, and actually it can be used for amelioration of acidic soils. In this case, biomass fly ash in acidic organically depleted glaze soil actually increase soil pH reaction, ameliorated soil acidity, improved nutrient content, but also and improved metal retention. Unfortunately, this matrix is very saline or alkaline, and in that case it increased soil salinity very, very strongly. Similar to other matrices like bottom ash and dolomite are also very, very common in amelioration of acidic soils. And bottom ash, like previous, can also be used as source of phosphorus, for instance, but in this case you can see that bottom ash actually increase soil pH reaction, increase content of available phosphorus and potassium, but also impacted in soil electrical conductivity. As very alkaline matrix, actually this matrix is enriched, of course, and with other nutrients like potassium, calcium, and magnesium, and in this case we notice that also it increased cadmium soil to plant transfer in radish plants. Okay, what solution we can propose? We can propose as solutions for amelioration of salt-affected and metal-contaminated areas. Some of widely used, for instance, are visible on this picture in one of the most intensive agri-ecosystems, Neretva River Valley, placed on the Adriatic coast. Actually, this measure is very important for the health and resilience of this intensive agri-ecosystem, which means regulation of groundwater level. It can be performed, like in this case, with pump stations, with over 20 pump stations distributed on 10,000 hectares area, and of course with open channels and underground pipelines. That is that measure was confirmed as very, very effective strategy for salinity areas. Another approach is application of appropriate, I would say appropriate, the best irrigation system. For instance, on this picture, you can see application of very, very modern both systems, but with different outcomes. For instance, application with same quality water, but with different irrigation system, like is micro irrigation, actually totally destroyed test crops, whereas in application of drip irrigation, but with relatively low quality water, actually we obtained quite good yield of test crop. Some other chemical amelioration measures assumes application of organic and mineral soil conditioners, as you saw on some previous slide. Then, of course, application of some nano-based solutions like nanomaterials, nano-based technologies, nanofiltration, and others. And, of course, spatio-temporal adaptation of cropping pattern. For instance, combination of different crop types like agroforestry, consociation of forage and cereal crops, and many others. Interesting that we heard from Judith's presentation also some of traits or even anatomical parts like roots can be transferred or directly used by grafting in horticulture. Grafting scions on salt-resistant rootstock can actually improve much of crops and improve their salt resistance. Exploitation of plant microbes association like symbiotic associated bacteria and arbuscular mycorrhizal fungi have been also shown as very effective strategies against salinity and metal contamination, because they can improve water absorption capacity and acquisition of essential nutrients. Breeding and genetic approaches, as we heard from Judith, are also very effective, albeit still in controlled, not in open field conditions. As you can see here, by transgenic approach is possible to obtain very strong and resistant genotypes, like in case of sugar beet or like in case of canola. But as I said, in lab, not yet in real conditions. Application of bio-based amendments, biochar, bioashes, and other microbial inocula are also possible in salt-affected and contaminated soils worldwide. For conclusions, some of mine are in line with Judith's and with Zed's. I will skip them. Also, it's important to say that managing of salinity types actually requires integrated approaches, which is a complex task for let's say, farmers. Various preventive solutions have been used in salt-stressed agri-ecosystems to improve plant plant production, but unfortunately with side effects, as you notice, for instance, with increased metal transfer in crops. Modern approaches like microbe plant association and nanotechnology show promising possibilities in alleviating salt and metal stress, but still need to be improved. And I would like to say that actually multidisciplinary approaches and solutions driven not only by plant and environmental scientists, but also included in other areas, remote sensing, artificial intelligence, machine learning, big data analysis, can ensure actually useful tools for detecting and protection of saline areas. Systematic screening of potential metalophyte can identify the most effective candidates for phytoremediation of metal contaminated soils. And of course, combine, as you saw, actually combine interaction among numerous pedovariables lead to multicollinearity, actually making specific and synergistic effects extremely hard to study. You saw some of double and triple interaction, but in real condition there are too many interactions that with still all those mechanisms are still unclear. And finally, due to possibility of fast processing and numerous predictors under different dose conditions, fast developing algorithms driven by AI appear to be promising approach to optimizing the management of metal contaminated and salt affected areas in different scenarios. Thank you very much for your attention.
Thank you, Professor Ondrášek, for your presentation. Now comes the question part. For the first three presentations you have heard, do you have any questions to the presenters? It's always difficult to make the first one. Let me the one. That's the task for the organizers. So, Professor Rengel, which is the most important source of phosphorus when you are talking about recirculation? Because I understood that the rock phosphate is a non-renewable resource, but what would be the most important renewable phosphorus resource in the agriculture? If you are not talking about farmyard manure, I don't know how much phosphorus in that. So what would be that?
It depends on the farming system. For example, in most farming systems, the easiest source of organic phosphorus are crop residues, and management of crop residues is hugely important. But for farming systems that are close to large cities, for example, wastewater, municipal wastewater, and sewage sludge are also hugely important sources of phosphorus. For example, in my case, Perth is the only large source of human waste, and agriculture is happening about 200 kilometers to the east of Perth, and that is too large a distance for transport of these organic sources of phosphorus. So in our case, farmers have to rely on crop residues. But in many other situations where cities or humans are concentrated relatively close by, in this case, municipal wastewater and sewage sludge would be excellent sources of organic phosphorus for recycling.
Thank you. That would make also an issue for the municipalities, because if we are also planning to use municipal waste for agriculture, because it's huge amount, not just nutrients, but also water. I have a calculation that we use as much liquid sourced from the human waste as we use for irrigation, the normal surface water. So it's a huge amount, but there are some issues on that, like multi-resistant bacteria and so on and so forth, that we have to also deal with.
Well, certainly there are numerous regulations that would have to be taken into account in terms of heavy metals, in terms of potential pathogens. So there is no solution that would be universal. It is case-by-case basis, so depends on the situation. You would have to choose the most appropriate measure.
Yeah.
Struvite is ammonium magnesium phosphate, which is a mineral that can be precipitated from wastewater, and it has been very successfully used as a recycled source of phosphorus for growing crops. But again, it is not applicable in every situation. You would have to have large volumes of wastewater to make it economically viable. So it works, certainly works, but whether it's economically viable, that's another layer of considerations that you would have to take into account.
Okay, thank you. Are there any? Yep, please. May I have the microphone? Okay.
I have a question for Prof. Zad. About your figure related to the biomass production versus the chemical consumption of phosphorus fertilizers. If you remember the figure about China, Africa, Europe, and USA. You have shown here that in Europe, nowadays, the consumption of the chemical fertilizer of phosphorus is going to be decreased, not even to stay as it is or stable, but decreased. Could you please explain what could be the reason behind this? Why, for example, in Africa it's increasing and China also increasing the consumption of the chemical fertilizers of phosphorus, while in Europe it decreased, while at the same time the biomass production increasing. So what could be the reason in Europe? Is it the agriculture policies, management, or a new type of fertilizers, or what?
It's a very good question, but a very complex question. I would need about 5 hours to explain it all. But you think I can do it? So let's just, the map that I didn't show you is about deficits and surpluses of phosphorus in soil. And if I had shown that map, you would notice that in Europe there is a very high surplus because of long history of relatively high rates of phosphorus fertilization. So there is plenty of residual soil phosphorus that can be reused. And modern genotypes are much better doing it than genotypes that have been bred in the past. Situation in China is slightly different. They also have very high surpluses of phosphorus, but they use the ridiculously high rates of phosphorus fertilizers because they were subsidized. So government intervention can skew up things very badly. And in China, it is so bad that eutrophication of water, if you want to study eutrophication by phosphorus and nitrogen, you need to go to China, because by far the worst examples on this planet exist in China because of government subsidies. Fertilizer is too cheap, and therefore farmers say, if 1 scoop is good, 2 scoops must be better. That's kind of human nature, unfortunately. So there are various aspects of utilizing of phosphorus that's already in there. You can claim that utilizing what's in there is also, because there is going to be a point in the future when whatever is, how large that pool is, it's going to run out eventually. So there are various aspects to this question. In terms of Africa, they have had very difficult time purchasing fertilizers because they were relatively expensive to them. Lately, there's lots more international intervention, lots more money flowing into Africa, trying to increase food production and make Africa self-sustainable in terms of food production, which is the reason that we are seeing modern genotypes, modern agronomy being applied to crops, and without necessarily increases in fertilizer application, we are seeing very substantial increases in food production.
Yeah, can we add also the soil type and the climate as a factor controlling this issue? I mean, because I come from Egypt and we have a desert and we have soil with pH above 8.5. So therefore the farmer not every year with every crop, because usually we cultivate like 2 or 3 times a year, not like here monoculture system. So we have to add too much phosphorus, but most of this phosphorus changed in the soil or turned in the soil into insoluble phosphorus. So therefore, we use always a high rate of the phosphorus fertilization, even higher than nitrogen or potassium, for example. So could the soil type, the difference between the soil type in Europe or in Africa, could be also a reason for the need to use phosphorus fertilization in the countries with alkaline soil?
Yeah, you are touching on another hours and hours of lecturing. As a very basic principle in acidic soils, there are iron and aluminium phosphates that are forming, and these are forms that plants cannot access. On the other side, with high pH, there are calcium phosphates being formed. But there are situations, and I was part of a very successful Chinese operation in northern China. There are also very alkaline soils. Using ammonium-based phosphorus fertilizers caused acidification of the rhizosphere soil and therefore solubilization of these calcium phosphates and considerably higher availability of phosphorus to plants due to agronomic practice, basically, change in agronomic practice. Very simple change in agronomic practice.
Okay, thank you. Thank you. So as we are approaching to the end of the first morning session, thank you for the presenters, for the presentations. Thank you for the questions. Now comes the coffee break, but before that, we have to make a group photo. So please stand up and come close, because our colleagues there will make a photo of us, and then because of the angle that he's having now, everybody will be seen. So please come forward and try to form a kind of group. So please take your seats. We are continuing with the second session. That's about animal production. So as I see the next presentation, which is a joint effort from our colleagues, they are also ready. So I invite Melinda Paulczek, Nora Vosch, and Levente Ceglidi. They will share the time and make the presentation, each of them talking about their specific topic. And the title of the presentation is Exploring the Interplay of Microbiome, Proteome, and Animal Health. Melinda, the floor is yours. Thank you.
Okay, thank you for the opportunity. And as you can see, I will start with the microbiome part of this presentation. So at the University of Debrecen, one of our main research areas is innovations, supporting the production of nutritionally enhanced agri-foods, quality meat with minimal drug residues. And out of the main challenges, one significant is that in the case of these intensive systems, animals are exposed to extreme stress, which renders their microbiota dysbiotic. Diversity will decrease, and this will manifest in sooner or later in chronic inflammation, and this exposes animals more prone to several infections and illnesses. So there is a dire need for solutions, and especially for green solutions to regenerate, to restore microbial balance and resilience. And in collaboration with animal breeders, veterinarians, and other scientists, we are investigating how the plant-derived biotic components, bioactive components can modulate microbiota and can support intestinal microbial health. To find connections between intestinal health, the health of the animal, and the production performance is not straightforward, because the expression of the microbiota-mediated traits depend on numerous factors, environmental factors, the nutrition, the genetic background of the host, and as you can see, these are interconnected. So investigating one thing or one aspect in isolation is not suggested anymore, since we have all the tools and the capacities to deep dive in the meshwork of these factors and the data. For example, as you probably know, potential pathogens are also part of the normal intestinal flora. However, they are not able to cause diseases until the beneficious microbiota don't let them overgrow. This is based on the competitive exclusion. And the flip side of the coin is, however, that in most cases, why probiotics are not efficient enough is that we forget to have a look at the already existing cultures, whether they are willing to accept the newcomers. You know, this is the new kid on the block concept. So one important area of our research is to characterize microbial communities, to reveal the patterns which make them good or bad responders to several treatments. And despite the vast amount of scientific data and results, we still need skepticism to find causations behind correlations. And since we are dealing with various animal samples, we needed a validated methodology, so we identified technical, analytical factors which can significantly distort the outcomes, the results, and we also know now how to avoid these biases as regarding, for example, the distribution of the gram-positive, gram-negative bacteria. And as you can see, during the past years, you can see the overview of our study protocols on farm animals, broiler, the broiler part, sorry. So actually above is the broiler, then the swine, and the carp at the bottom part. And you can see that for the positive control, we have applied beta-glucan, and you can see that we treated the animals with either anthocyanins, fructooligosaccharides, carotenoids, some symbionts, and the combination of these were added to their forages, and these bioactive compounds have been extracted from the food waste. So there is a kind of recycling in this. It's a kind of green approach. And among others, we investigated microbial growth promoters, growth inhibitors, and we also investigated how the diversity is changing throughout the production period. And what is very typical, and you can see that with the age of the animals, diversity constantly increases, and around the end of the production period, there is a steep decline in the diversity, which is also accompanied by a steep decline in the health status of the animals. However, in the case of our treatment groups, as you can also see, there was we have seen that these phytonutrients have a kind of protective effect against the deteriorating parameters, so they might be good options in restoring the microbial intestinal health. And for me, it was very interesting to recognize how well organized biological systems are. A good example from our everyday life is the flocking of starlings in the sky, when you see these beautiful patterns, and in these cases, every bird knows where and when to turn to make these mesmerizing patterns. And the thing is very alike in the case of balanced microbial communities. And we are mostly interested in the tendentious changes of the communities. Do they become, the community architectures, are they becoming more hierarchical or more random? What about the hubs? What about the keystone taxa, which are responsible for maintaining the health of the intestine? And during the past years, we have developed a service portfolio to investigate the intestinal microbiota of farm animals with regard to diversity matrices, probiotics, short-chain fatty acids, microbiome growth promoters, inhibitors, in the case of the flock, the abdominal fat deposition, and the microbiota-mediated amino acid, vitamin metabolisms, and so on. And we are living in a huge crisis regarding antibiotic resistance. One important reason is purely economical. So you probably know it, developing drugs for short-term infectious diseases is far less beneficial than developing drugs for long-term chronic illnesses. And second, 80% of the antibiotics developed are used in agriculture and have been used for many, many years as boosters. So considering the genetic factors such as horizontal gene transfer, public transport, and the global food chain, the antibiotic resistance genes, they spread very rapidly. So there is a huge responsibility on this sector. And as we have also seen in our data, you can see we compare the environmental pollution with regard to the environmental pollution. On the left side, you can see human intensive care unit samples and the environment, and on the right side, the flock samples, stool samples, and the environment, and you can see that the contamination is much better in the case of the animal meat production. And we may not forget that about 70% of the antibiotics used in the agriculture are also used in the human medicine. So we are basically interested in the investigation of resistant gene accumulations in the environment and the possible transmission routes to support decision-making and stewardship. And thank you for the opportunity.
Thank you very much. I continue with the second part of this presentation, and that's a very specific topic in animal science. It's called animal proteomics. What could be the role of proteomics in One Health? Animal disease and zoonotic infections are one of the major concerns today. Proteomics can be used for the diagnosis, the pathogenic pathways, and also for vaccine development. Antibiotic resistance was mentioned that it's a huge problem today, and the discovery of novel treatments by the tools of proteomics is an emerging field. For animal production as a production system, food adulteration, detection of allergens that could be considered in case of cow milk, or some contaminants which could be for living animals or during the processing, could be the field where proteomics can be applied. What is this approach? What is proteomics? In case of proteomics, we are looking for all the proteins expressed in a tissue, in a biological tissue, at a certain point. If we focus a little bit on my field in animal production, proteomics could contribute to a successful biomarker discovery. Genetic factors, and to identify genetic factors, is a widespread possibility today, and we have the option for an individual to have a look at 700,000 genetic points in the genome. In case of proteomics, somehow this is also inherited, but in most cases, this is connected to the animal physiology. What are the fields? Animal nutrition, environmental factors, management, transport, slaughter, and animal health. Stress and welfare factors could be also identified with proteomic tools, and the result, a more efficient and better animal quality, more efficient production and a better quality of product. Two types of set of tools can be used in the laboratory. The top-down proteomics, that's the earliest one, where the separation is made by gears. It could be one or two-dimensional and different approaches, and target molecules are identified by mass spectrometry. The other one is bottom-up proteomics. That's the state-of-the-art methodology where the number of unique molecules is much more higher. And the result is the identified protein, which is related to a trait with economic value. At the department, in our laboratory, we are using the top-down method, where we are focusing different separation techniques, and the result that in a type of tissue we are able to work parallel with approximately, let's say, maximum 1,000 of unique protein spots, and searching for the spots which could be the biomarker of a certain trait. This is a very nice example from a study how these tools can be used. So this study was conducted on Rouge de Pré breed in France, and they were looking for the housing and nutrition background, and they have seen that the grass, haylage, hay based on foraging could influence the carcass quality and the meat quality. We know how to feed the animal, what type of nutrition and what kind of animal genetics resulted in what level of yield and what type of quality parameters. But using proteomic tools, we have the option to know what is the molecular background, so what is happening in the animal. Some years ago, we conducted a study where different slaughter weight was the focus. We used Charolais bulls, and these bulls were slaughtered at 5,000, 6,000, 7,000 kilograms of body weight. We know that different age and different body weight is strongly connected to quality parameters, and we know that what type of meat quality is expected, and using these proteomic tools we were able to identify some set of proteins which are related to better quality, higher price. For example, the tenderness was identified that is connected to heat shock protein. A very similar study conducted later by Baudon et al. In 2020, and they were focusing on the same traits as we did, but they were also looking at the plasma proteome level, and the result of their study and our study shows that there is a major overlap of biomarker proteins, so it was a type of confirmation what we found. There were some studies where we were focusing on the pituitary adenyl cyclase activating polypeptide, which has a very essential role in the cell function. And also it has some role connected to economic traits like girth size, body weight, behavior of the animal. And we were the first group who recognized or discovered that the yield of this peptide in the milk is very high. So we were looking for some additional factors which are related to this peptide level, and also the role in the infant. And then, using again the proteomic tools, we were able to identify proteins which were related or which are related to this PCAP peptide. We divided our Holstein cows into two groups. One of them were high PCAP level, others has a low PCAP level in the blood plasma, and we successfully identified proteins which are related to these functions. There were another study conducted on rodents, and they were also investigating this PCAP molecule in the central nervous system. They administered a level of PCAP, and using proteomic tools, they were able to find some molecules which are closely related to PCAP level, confirmed by Western blot, and they found this CRMP2 molecule that it's a strong controlling factor of PCAP-regulated control in the brain. And what is the aspect for this rodent study to animal science, that this protein is proved to be very strong connection with mad cow disease. And the next presenter is talking about the animal health diseases and the possibilities how to identify or diagnose them. Thank you.
Thank you very much, and as we are almost running out of time, I have to be very, very quick, so maybe I will leave out some things. So out of the big topic of animal health, we had to choose a certain area, which is parasitology. Our institute is dealing with reproduction and parasitology also, that's why I have chosen this small area. Although it is very, very important in animal health, we are facing a lot of problems in connection with parasitology also. All farmed animals, all ruminants who are grazing are exposed to different internal parasites, and as you see, they can negatively impact a lot of things in animal health and in production. And as in the case of antibiotics, we are also facing a lot of resistant issues, and also we don't want any residues in our food chain. That's why we really, really have to think of the accurate diagnosis and the alternative treatments also. The easiest and sometimes the cheapest way to treat the animals with different drugs, but there are already several strains of different worms who are already called pan-resistant, so we have no effective drugs against them. I will talk about that later. On the picture, you see the clinical symptoms of the most important worm in small ruminant breeding, which is called Haemonchus contortus, and it causes severe anemia in the animal and so-called bottle jaw, which is caused due to the edema. Microbiome has a very strong effect on parasite infections in the animals. We know that already, although they are just recent studies from the last several years. We know what is microbiota. I'm not going to explain that. And microbiota has an overall effect on the humans and animals' body. Macrobiota, the parasites, are inside the host. And there is a so-called metamicrobiome, because scientists already started to examine the parasites' microbiota, actually, the microbes that are inside and on the parasite itself. And without being comprehensive, I'm just telling you some researches. For example, it was very interesting for me, the first one that has been conducted with mice. There's a worm so-called Heligmosomedes polygyrus, and the infection of this, if we increase the population of this worm, it will cause the rise of the population of lactobacillus in the mice's gut, which is a beneficial effect actually, so we wouldn't wait for that. And on the picture on the left, you see this human hookworm, and we have several new findings in connection with that also, which is in connection with the appearance of autoimmune diseases in humans. I already told you that resistance is a huge problem, and we are facing, even in Hungary and all over the world, huge resistance problems around sheep flux, and every day we are fighting against this disease. Haemonchus contortus is a highly pathogenic worm. You see it on the first picture, and it causes huge problems in sheep industry. And here in Hungary, we have several multi-resistant strains, so we really have to think of how we are treating. And for example, in the United Kingdom, there are pan-resistant strains also. And it has been found out that scientists started to examine the microbiome of these worms, and Prevotella and Fibrobacter species are inhabiting the gut of the worms. And interestingly, E. coli is much more the natural colonizer of this worm than, for example, in humans or in other mammals. And it will, or it maybe will lead us to find out a specific drug development, which will be nematode specific. But of course, it needs further research. Proteomics is very, very important in parasite diagnostics also. We heard a lot from my colleague about proteomics, because we need to give accurate diagnosis in connection with the parasite infections. And some examples for that, we are able to examine the secretome, the protein secreted from the gut or the cuticle of the host in connection with Haemonchus contortus infection. We are able to find extracellular vesicular proteins in the host, in the case of Ascaris suum, for example. And in connection with the most important Haemonchus contortus infection, the entire protein profile is already discovered. And just one interesting detail, you see Ratska sheep, our indigenous breed in Hungary. And we know that indigenous sheep breeds are less susceptible to parasitic infections than the culture breeds. And we are able to examine the proteome, the proteomic background of this, and we can use later in breeding. And as I see our dean, just a last few words about our experiments in the laboratories, in the institute. We are using for parasite diagnosis the conventional diagnostic tool, which is the classical taking fecal samples from the animals and checking the larvae and the eggs under the microscope. There's already a semi-automated tool for that also, and we use molecular tools in parasitic diagnosis also, which can be PCR-based. And our new research, which has been published in veterinary parasitology and parasite also, that we have found out a new protocol for an on-farm diagnostic tool, which is a loop-mediated isothermal amplification-based, and the other one is the recombinase polymerase amplification-based protocol. And it's important because you can take it out to the farm easily, and by a colorimetric change, you can see the positive and the negative samples, and just quickly showing you, it's like a pregnancy test. You just take it into the sample, and it will tell you if it's Haemonchus contortus or not. And this is all the devices that you can take out to the farm. And next to the animals, you can tell the owner that what is the next step in the protocol. And thank you very much for your attention.
Thank you. It's always a difficult task to show that the time is about to end. I know 20 minutes is not enough, especially if you are talking about a complex topic as microbiome, proteome, and animal health. But one intention of us is just to show you what things that we are up to, and then just to, I hope that we can put together this conference a information package. I will talk to the presenters if we can take some information from the presentations and spread it among the audience. Okay, so as the next presentation in this animal topic, may I ask Professor Norbert Lukács from Slovak University of Agriculture to give the lecture named Potential Ability of Endocrine Disruptors to Affect the Living System, an in vitro study. Professor, the floor is yours.
Thank you so much. I firstly thank you, Ode Laszlo, on the invite on the conference. Ladies and gentlemen, I would like to present our results with the title Potential Ability of Endocrine Disruptor of the Effect to the Living System and vitro study. So I'm working in the Slovak University of Agriculture Institute of Applied Biology. Some words of the introduce during past 50 years and most 1,000 metals and plastic chemicals have been released to the general environment, and some natural and synthetic chemicals can interfere with normal function and endocrine systems, and these chemicals and released to the colon of the endocrine disruptors and use of the appropriate ADC. It is a very important picture because in the most harvest of the waste content of the mainly plastic, and nowadays we talk about the plastic age from the 1915 to the present time. We live every day with the plastic material and use for the everyday different kind of the plastic. Global production of the plastic and every year and then more and more and higher and higher, and to 2021 was slightly 400 million metric tons, and distribution in the area and the main production on the Asia and also US and the North America and also in the Europe. Many plastic material used for the daily life and from the polypropylene, polyethylene, polyvinyl chloride in the packaging and healthcare and building industry and etc. and different. This is also very interesting. We know about the flooding of the island from the plastic, and the area in this island larger than Spain, and smaller 4 and 5 smaller plastic island also floating in the Pacific. These endocrine disruptors and the typical chemicals and the pesticide and the pharmaceuticals and the industrial chemical, I focused in this presentation mainly to the bisphenols and nonylphenol and octylphenols, and bisphenol used in the mainly of the most popular of the bisphenol A into the packaging and the plastic containers and canned foods, cosmetics, electronic devices, and sport equipment. And next, for the pathway of the function of the endocrine disruptor and the different 3 parts and the mimic of the natural hormones or blockation of the natural hormones or modulation of the natural hormones, mainly of the target cells, and some exact mechanism in the different cells, and very wide. In the presentation, in the work and the mechanism, the 3 parts, and one of the binding of the mainly of the estrogen receptors, other side, and focused to the generation of reoxide oxygen species, and also we know about the effect of endocrine disruptor to the intracellular connection and communication between cells. According to Young determined the difference classification of the endocrine disruptors and the mainly groups and the pesticide, organophosphate, alkyl phenols, and from this alkyl phenols I focused to the nonylphenol and octylphenol, also heavy metals and phthalates and last but not least, in the phenols and the phenol A, and then nowadays many usage of the replacement of the bisphenol F or bisphenol S, bisphenol B, and other. So I told about to the presentation. I focused to the effect to the 4-nonylphenol, 4-octylphenol, and mainly to the bisphenol A and S, and experiment and the results from experiment and the partial experiment from the determination of an effect of the nonylphenol to the activation of the receptors on the progesterone receptor on the spermatozoa. Next of the effect of nonylphenol and octylphenol to the produce of the steroid hormones from the cell lines and the Leydig cell lines. Next on the effect of the bisphenols to the cell viability and steroid hormones produced in the human adrenocortical cell line, and the last in the immunomodulation or effect of the processing of bisphenol to the inflammation process in the HUVEC cell lines. In the nonylphenol, we detected in the progesterone receptor in the spermatozoa and very good cells for the detection of different receptors, mainly GnRH, LG, mainly of the androgen and progesterone, and we analyze in our labs in the one part of the motility, and then the second part was analysis of the fluorescent detection of the progesterone receptor positive parts, and you can show this picture. It's a negative control without any fluorescent lights in the 1A and the 1B and the formation of the head of the spermatozoa. And if you in the positive control with the added of the progesterone, and if you added of the different concentration of nonylphenol with a higher count of the positive place and the positive post-acrosomal part with the progesterone receptor in the 10 micrograms of nonylphenol in the 17.87%, and if you high concentration and these positive parts more than 21% of the positive. This conclusion is the detection of the higher concentration of nonylphenol, increase of the binding affinity of progesterone receptor in the spermatozoa. Next research was focused to the different parts in the Leydig cells and the effect of the nonylphenol and octylphenol, and viability and oxidative status, production of the hormone and the steroid hormones, and assay of the quality of the membrane and the membrane integrity by the detection of fluorescent detection of the connection and the condition of the connexins in the cells. In the chart we show effect in the first line and the up line in the nonylphenol and the down line in the octylphenol, and compare it with the control and the higher concentration up to 20.5. It's a positively and the significantly correlation and the production of the hormones and the steroid hormones of the Leydig hormones and typical of the androstenedione and testosterone. If we compare with the mitochondrial activity or viability of the cells, we detected also higher concentration, 1 or 22.5. It's a positively effect of this chemicals in this nonyl phenol of the octyl phenol to the mitochondrial function. In the analysis of the reactive oxygen species by chemiluminescence methods, we analyze it in the lower and on the all concentration with observed activated of the higher concentration of the higher produce of the reactive oxygen species. Compare of the connection and the condition of the connection of the cells connection. If you compare after 30 minutes incubated with the nonyl phenol, octyl phenol, we detected also in the higher concentration from 5 pounds and in the octyl phenol more than 25 micrograms activated of the dysfunction of disruption of this mechanism and the negatively effect to the connexin condition. After 24 hours, and these signs will be highly and positively negative effect to the connection of the cells. Next part was analysis of the bisphenols and the bisphenols to the adenocarcinoma cell lines. In the cell lines and good use for the analysis of all steroid hormones, and also detected of the cell viability, membrane integrity, and the oxidative status. And we compare on the different bisphenols and the bisphenol B, F, AF, A, and S. And on the chart we show down many results, and basically more than 1 micromole of the bisphenol A, also bisphenol B, bisphenol F and bisphenol S is activated of the negatively effect of the mitochondrial function. And if you compare with the toxicity and the higher cytotoxicity of the B, compare with the S, with the very low effect of the cytotoxicity. We detected of the function of the integrity of cell membrane, also and the similar data like the previous data. And the steroidogenesis in the complex process where from the synthesis of the steroid hormone, different hormone from the cholesterol, and the basically compounds for the different steroids, and the main function in the processes are of the enzymes. And if we detection of the last steroid hormone from different tissue of the aldosterone, cortisol, and estradiol. Shortly, brief conclusion about the effect of different bisphenols. Bisphenol A affects to the concentration of the progesterone. Bisphenol AF affects to the cholesterol. Bisphenol B affecting to the concentration of the estradiol. Bisphenol F to the testosterone, and bisphenol S to the cortisol levels. And then nowadays we focus it to the investigation of the effect of endocrine disruption to the inflammation process and the process of the immunological process. And we know about to the inflammation and the basically pathological process and defense mechanism from the destroying of the organs, and the typical signs of the inflammation of redness, swelling, head pain, and the loss of the function, and the signs of the microorganism. If you compare and for the models of the analysis of inflammation process of the microenvironment in the endothelial cell or vascular parts. We know about to the different molecules from the membranes, phospholipids, according to the across to the different way, cyclooxygenase 1 and cyclooxygenase 2, and activating of the specific synthesize of the prostaglandins, and activated of the immune system and the produce of the different cytokines, interleukins. This is only partially result and the effect of bisphenol S to the UVEC cell lines. UVEC cell lines, it's a model for the human vascular endothelial cell line and the typical cell line for use on the process of the inflammation. If you compare of the basic concentration with the control and positive control with the bisphenol S, we know with the viability of these cells and the other metabolical activity and significant signs, but if you detection of the cytokines and the interleukin 6 and concentration more than 1 micromole, it's a higher concentration and the higher production of the interleukin 6 and also activated of the inflammation process from the prostaglandin 8-2. Conclusion, we know that bisphenols is a dose-dependent effect and affecting of the mitochondrial activity, integrity of lysosomes, integrity of cell membranes, also affecting of the oxidative and antioxidant status through to the generation of the superoxide radicals and activation on the affecting of the produce of the basic enzyme of the antioxidant enzymes of the superoxidism, the glutathione peroxidase. And I told about the cells secretion activity and affecting of the biosynthesis of the different steroid hormones and also slightly decrease of the production of the interleukin 8 to the endothelial cells. Thank you so much for your attention.
Thank you for the presentation. It's always good to see that dealing with the endocrine system and studying the hormones is a very difficult task, because there are almost unknown interactions between the different parts of our neurohormonal system, and if you are interfering in one, that you don't know what can you cause in the other end. So that was very interesting. Thank you for that. Now we are moving forward to the environmental part of this One Health program, and two topics in connection with water. That's one very crucial element of our environment, actually basic of life. That was my professor told me in my early studies. Water is the most important element in the world, the basics for the rest of the life. That's it. So on this topic, may I introduce Professor János Józsa from Budapest University of Technology and Economics to present you on current multidisciplinary challenges in water management, selected Hungarian case studies. Professor, the floor is yours.
Thank you very much. So good late morning, ladies and gentlemen. I will speak here on behalf of Budapest University of Technology and Economics, and in particular the Department of Hydraulic and Water Resources Engineering. I will present some of our achievements in surface water environment, mainly within our expertise, which means the physical part of the processes. But as those water bodies are habitats for the biosphere, we believe that our talk and then our messages will be very much useful for the kind of biological community and the agro community. Our past cooperations all prove that. So we will start with a kind of fluvial analysis, and then we will take a look first at the whole Danube catchment. I mentioned that we would discuss different scales, from the catchment down to the kind of point scale, I mean single gravel particle on the bottom of the river. Would be fine to kind of erase this red one, but I may then ruin something. So why scale-related analysis, downscaling, upscaling? So the hydro and morphodynamic processes occur over a wide range of scales, from continental, regional, to the initiation of motion of individual particles. That is the point scale. Of course, smaller scales and smaller scale processes are usually embedded in larger ones. This is the so-called opportunity to carry out then the so-called downscaling and upscaling of the processes and to see all the kind of important interaction mechanisms. Now turning to biology, biological processes also span different and reflect specific scales, all key to biodiversity, if it's rich or poor. Strong interdependency and interaction between the varying scale occur, and this gives the importance to deal with that from a scientific point of view. First, let's see the largest, the so-called catchment-wide scale, more exactly the River Danube catchment. In a recent international project, we established the kind of sediment, suspended sediment balance for the whole river system, for the period, based on data from the period before the large-scale hydropower constructions, and then also from the data collected these days. You can see the major difference between the two. So on the left-hand side, you have the kind of historical ones, and then on the right-hand side, you cannot not to see the impact of the hydropower plants. This change is tremendous, and its impact can be just imagined on the biosphere, the fauna and the flora in the catchment. There are ongoing studies to try to improve the conditions, which are quite poor these days. Very strong consortia and working on creating some new techniques, new methodology on how to improve the conditions, all under a kind of conversation of hydrobiologists. Anyway, so this was a kind of almost global scale. And now let's make a tour from that point of view, a kind of environment and surface water environment over Hungary, which is in the Carpathian Basin. As I mentioned, I will just stay within our expertise, which is hydrodynamics, morphodynamics, and thermodynamics, as well as habitat issues. First example will be the so-called Szigetköz floodplain and secondary branch system of Danube in northwest Hungary. You can see the red rectangle indicating the area for that particular case study. There it was interesting to see, also to kind of evaluate the biosphere and habitat conditions, the flooding conditions and the rate of flooding of the system under different flow rates. So here you can see side by side 3 different discharge, which is the flow rate, a kind of more than mid-flow, and a kind of mid-flow, and then smaller than that. You can see the clear difference in the flooded area ratio to the complete one. Let's now go through step by step on a kind of sequence of this flooding process, starting from a very high flow rate. You can see that all the area is safely flooded. Then I reduce the discharge. You can see that it became shallower, but still kind of globally flooded, still even shallower. Then it's already just partially flooded, which makes a big difference for the fauna and flora. There I go even lower, very much reducing. And then finally a rather low flow rate, which shows that there are only very limited particular areas that are inundated. All that is a key issue to quantify there the habitat conditions, depth, velocity, and interconnection of the separated or non-separated flow bodies together. It's really a key for a better and improved qualification, and gives some basis for any restorational studies and planning. Let's move further on. This is just one example for a rather low flow rate. On the left-hand side, you can see the velocity distribution, and on the right-hand side, you can see a kind of distinction of sustained flow, intermittent flow, and stagnant water sub-areas. These are, of course, very different for the quantification, qualification, and the classification of the region from a habitat quality point of view. Let's move to the south part of River Danube, which is a major river in Hungary, and let's say the so-called Gemens floodplain and secondary branch system, in a way quite similar to the one discussed before. And it is, as I mentioned, it is in South Hungary, very much complicated. The main channel and then the secondary branches and the floodplain. So what we carried out here is to use a series, a subsequent series of different flow rates, and see the rate of inundation there, and again quantify it even statistically from a depth, velocity, and connectivity joint point of view. So I will start from a very low flow rate. On the right-hand side, you can see quite closely one particular part of the system. There is flow only in the main channel. Increasing the flow rate, further increasing, we are already at 3,000 cubic meter per second. You can see some secondary branches flooded, so made active in the system. We step onward further on, already more than 4,000, even more, and then finally we are at the top, and we can see that most part of it are inundated, and there are still kind of dry islands. So this is just a pattern to have a feeling on the complexity of the flooding of such area, and to use it, to make use of this information when evaluating the biosphere, the biodiversity there, not to mention when there are intentions to improve all that. third example will be returning to the north part of it, local scale. So we have the reach scale up till now, but now we go to a local scale. The Rab-Mosoni-Danube rivers confluence in northwest Hungary. What I'm going to speak about is to show the complexity of mixing at such a confluence. I use a kind of unfortunate tracer study, the red mud spill in 2010 in Hungary, which reached the natural water courses and arrived even to the town of Győr at this confluence, luckily already neutralized by plaster powder, so harmless and giving the room to understand these complex mixing problems to us. Try to find the, oops, not? Okay. We carried out three-dimensional turbulent simulation of the process, quite nicely matching the observed patterns. There you can see the combination of the von Karman vortex shedding. Von Karman was graduated at our university, by the way. The so-called Kelvin-Helmholtz instability due to the strong velocity shear in a local scale, and even the secondary flows. All that makes this process locally very, very complicated. This kind of, as I mentioned, unfortunate disaster case helped us to have a much deeper understanding on all this, which we can then utilize later on in other cases. Nowadays there is no presentation without showing the utility of artificial intelligence. Underwater video camera movie on a gravelous bed surface, and the artificial intelligence tool was able to learn how to distinguish subregions with different grain size. So what was showed is the kind of work by the artificial intelligence, kind of, I would say, very fast, once it has been taught on that. Let's move on to go to a local even point scale. These days we are now able even to simulate the motion of the fish. I am sorry. That way. And fish motion together with the water motion, the so-called Lagrangian smooth particle hydrodynamics is utilized in that, and we can go even to fish path, fish ladders, just to see a bit more in details the behavior of fish and to make the conditions easier for them to take the way up the stream. At last, in this series, we can see a kind of point scale, again underwater video and the motion of single gravel particles, and we can reconstruct and understand and quantify their motion, even their velocity based on the particle image velocity based analysis, kind of image processing, which is very up to date. I'm just showing to you just to see that it is feasible by now. Let's go on. Let's move to the middle part of River Danube and see the cooling water jet behavior produced by a nuclear power plant at Paks region. You can see the location of that. Let's move on. This is an aerial photo. So this is the, this is the cooling water, already warm water outlet. This is the jet, and this is the drone we used, supplied with video, classical one, and also thermal camera to see the process more in details. Just the normal camera movie. Right. It's a bit speeded up, but still quite slow. So you can see that you can see something, but you cannot see the behavior from a thermal point of view. So for that, let's move on and let's see the movie taken by the thermal camera. Very good. Jet is seen on this side. And what you can see that instead of a kind of smooth plume, according to the Gaussian normal distribution function, we can see a kind of fingering of the plume, which is also valid for the atmosphere. So cyclones comes and then split it into fingers and then go on by that format. And this is alarming from the point of view of the quantification of all that, as if it's a kind of harmful or harmless effect on the habitat nearby. And this kind of non-homogeneous character might be kind of enough reason to revisit the environmental prescriptions for that. Anyway, it's good to know, and good to know that there is already technique available to take that. Let's move to lakes. Lake Balaton, known possibly by everybody. This is a very large, shallow lake, the largest shallow lake in Central Europe. We had a very strong cyclone, the so-called Kirill one, coming from the southwest. This is several days. And motion was induced by that on the water surface and, of course, also inside the body. This oscillatory motion is a kind of back and forth motion like this. And you can follow that on a replay of a two-dimensional hydrodynamic simulation. We just started, and there are two kind of hydrographs or limnigraphs, and then the kind of strong period will just come. It has arrived, 100 kilometers per hour, up, and then stopped. And then you have the backward motion. This is just to show that it's a kind of not very inhomogeneous topography, but we have Tihany Peninsula, there Tihany Straits, and it kind of nearly cut the behavior into two parts, but there is an interaction at the strait, so complex enough to deal with it with sophisticated hydrodynamics tools. Let's move on. And let's see some patterns which show even a fractal geometry nature. This is a recent algae distribution, blue algae in Lake Balaton. You can see patches, you can see filaments. So it shows that, of course, there is the blooming, but there is also the drift by the wind-induced currents, and it makes the picture very much complicated, which is important to know about. So you cannot really wait for a kind of exact, well-forecasted behavior and pattern. You have to deal with the character of the pattern, and you have to look for similarities between the patterns. At least let's move on. There are some snapshots of this kind of chaotic fractal mixing patterns in different waters. And then this is a kind of ancient satellite image on Lake Balaton with some infrared spectrum, which shows in a windy situation the development of the suspended solids concentration pattern, which shows really very strong filamenter pattern. So we try to reproduce it by computer modeling, the hydrodynamic behavior, hydrodynamics and sediment dynamics together. No, the wind is reluctant to blow today. Let's move on. We didn't really lose too much. What I'm going to say that because time is running like hell, that I wanted to go on to the kind of point scale analysis, but there is no time in it. However, luckily it was done, such a point-like analysis in the early 60s. So that would be myself. So you can see my character turning toward becoming a scientific researcher, and my beloved mother keep an eye on me to be instructed enough on that. So a kind of very, very peaceful, comfortable time at Lake Balaton in the early 60s. Let me move on. Let's go to another lake, Lake Neusiedl, Fertő in Hungarian. This is a kind of aerial view of the lake with its surroundings and the prevailing north-northwest wind. And then the issue to have water supply to the lake comes up from time to time. So this is a simulation in 2 days, windy conditions, kind of water supply applied at this location, let it into the lake, let it go into the lake, discretize it into water parcels, and then drifted by the wind-induced motion, oscillatory combined with circulatory motion. And this is enough, it's not very complicated, but enough to result in a kind of fractal geometry behavior. I just hope that the... Okay, so time is running. Already 1 day, there will be several days. So the hope in such a case is the fastest possible homogenization in the receiving water body. It does not look homogeneous at all, so it's kind of fractal character can be shown and can be seen surviving for a very long while. So it's again alarming for the planning engineers that a proper choice of the point of release for the supply water and proper time waiting for a kind of sufficient mixing is needed, and these kind of limits have to be taken into count into expectation in the change of the biosphere. Of course, scales are very different from each other, and you have to aggregate the things. Last but one example, I came a bit closer to Debrecen. This is our second largest river Tisza, Theiss in German, and there is a kind of dammed area, and there we call it now Tisza Lake, a kind of reservoir, very large reservoir, more than 50 years old. Very nice biosphere there. This is the complexity of the topography, and we carried out hydro and morphodynamics calculations, two-dimensional ones, and this is the kind of flexible gridding to take into account the kind of local irregularities even of side branches or channels. So let's move here. Let's apply opening of some flushing channels, its intrusion by a suspended sediment plume, and then all of a sudden the arrival of the north wind. So this is okay, and then the wind will come, and as you can see, it will drift substantially the plume. So again, a phenomenon which has to be taken into account in a more complex study. Happily, the last example, we go to the space and make use of data produced by satellites, MODIS, US satellite for the time being, and we are going to make use of the data of that on estimating the Earth surface evapotranspiration for all Hungary. Here I just set the essential points. So evapotranspiration and surface temperature can be related to each other. There are physical evidences by which we can set up a transformation model from the surface temperature produced by the satellite at a very high resolution, 1 kilometer cell size, which is very promising, and it's also very kind of dense in time, and we use the so-called CREMAP model to produce the mapping of the evapotranspiration, which can be then used for several goals. So this is the established, detailed and accurate evapotranspiration map for Hungary. Of course, the highest over large open water surfaces, and also in the forested regions. These are almost evidences, but this one for the whole country is a very useful information anyway. Then also we can determine the rate of the mean annual evapotranspiration and precipitation, which means that we can judge if there is a kind of positive or negative recharge to groundwater as a balance of the, or as a kind of comparison of precipitation as input and evapotranspiration as loss. Here you can also see that it is irregular enough to make the whole analysis worth doing. Very last slide would be on the Danube-Tisa interfluvial region. Again, the precipitation minus evapotranspiration map and distribution. Again, what I'm going to say that it is irregular enough to be worth producing, and also can serve different important goals. Large-scale irrigation planning, then the before-mentioned cooling water conditions of the receiving Danube, especially at low flow rates, and also the wet habitat evaluation and even restoration for birds. So that was the kind of rather fast tour, kind of forced by myself. It was instructive, at least for myself, the producer of this presentation, but I hope that you could also enjoy that. So that would be the end, and thanks for your attention.
Thank you for this interesting and very nice presentation. It's easier to, when you use the computer-aided presentation and that kind of tools, that it will make us easier to understand how the biosphere and how river corridors operate. That is also one example that the holistic approach of our studies in the life sciences wide interconnected. I'm a bit sorry about this noise in the back, but we just started the education and our students are full of energy since they took their holidays but let's not move away from our streamline. May I ask Professor János Bogárdi, University of Bonn, to make the presentation, the title, One World, One Water Cycle, One Health. Examples how to tackle this global challenge.
Thank
Professor,
you very much. Thank you for the invitation. It's a big pleasure to be here. I praise the same hall where almost 30 years ago I was given the honorary citizenship of the then Debrecen Agricultural University. I changed the title of my presentation for a little bit because I knew that I have only 20 minutes, and in 20 minutes I will deal only with one example. And I'm very grateful to Professor Józsa, who showed in different scales what is happening in a water body, because I am going to concentrate on a continental scale or a part of a continental scale, not going into these hydrodynamic details, what is happening in every sentence I am saying. Beyond that, you have all this sequence of hydrodynamics. And also, I want to link all what I am going to say about water to societal needs and why we need to talk about water. And I start somewhere quite far away. It is the last 100,000 years of the Earth's history, and you see that there are only in the last couple of thousand years, not more than 10,000 years, when the climate suddenly became very timid, enabling us to use agriculture, to develop the civilizations first and foremost along the big rivers. And this almost anomaly of the Earth's history, what we want to sustain. Before that, we had 90,000 years of rough ride with huge migratory movements lasting 20, 30,000 years, when out of Africa humanity colonized the world. So this is part of our history. Migration is something which is happening irrespective of the connotation which we have these days. We are talking, if you ask people what is the biggest problem of today, you would always hear climate change. And I challenge this statement because climate change, irrespective that it is a serious matter, is only part of the challenges, and it is a consequence of much more fundamental challenges we are basically ignoring or not discussing. If you talk about climate change, it is something, there is a big fire and you are concerning about the smoke, but you forget to extinguish the fire. Now, the problems we are facing are always starting with P, population dynamics, which means growth, unregulated urbanization, migration. It is not forecasted, not dealt with in a way it should have been politically and scientifically, and it is always happened and it is happening. The second is poverty or the uneven distribution of wealth, and also opportunities between nations and different social strata, and it is getting worse worldwide. And the third one is pollution, where also climate change, which you can characterize as an atmospheric pollution of thermal and chemical origin, is belonging to, and this is accelerating. We are talking about climate change, but the CO2 content of the atmosphere is still increasing. And all these three major problems we are facing on global scale, on regional scale, on continental scales, there is not a competent global governance scheme nor coherent concepts how to deal with them. We are heading big trouble. And the human population says, there is a very colloquial say that half of humanity, whoever born in this planet, is still with us alive today. It means, you know, that this is an accelerating growth, even so if the acceleration is a little bit reduced by the now. If we look into the world today, you realize, and I'm listing here only problems which affect at least 1 billion or plus minus 1 billion people, and I give also in italics, in blue color, to refer to water, what are the water resources management implications of these problems. I do not read it out while I'm talking. Please look into these entries. I concentrate on two. There are still around 1 billion subsistence farmers in the world, and there are also about 1 billion slum dwellers in the world. And these two communities are not the same. So it means there are at least 2 billion people, if not more, who are living under conditions no one would consider to be human or dignified in the 21st century. So in a very simple arithmetic, we can say that about 1/3 of humanity is living under conditions none of us in this room would ever want to experience. And I almost challenge you all, probably none of you have even seen these people in their life. we tried to solve these problems. In 2015, the United Nations discussed and adopted the 17 Sustainable Development Goals with 169 target values. And as a water resources engineer, I always use this unofficial display where the sixth goal, the water is in the center of the gravity. It is a capstone, or it is the X around which development should rotate. But unfortunately, SDG 6 is in grave delay. We already know that we would not achieve at least this objective, and probably many of the other objectives will fail to be achieved. So we agree, we vote for it, but we don't vote money for it, and we basically do not tackle it. This is big negligence. Now, especially when we now try to come closer to water resources management, you realize that if you take some countries or parts of continents, you realize that economic or human well-being and the availability of storage space to store water, to secure water supply, to secure water for humans, for crops, and for animals is closely related. Australia and Northern America has the highest per capita storage capacity, and these are among the most attractive continents to live there. And you see that some of the African countries, even the partially well-developed South Africa or even Ethiopia, are very low on per capita storage volume. Even if the highly contested Great Ethiopian Renaissance Dam would come into operation very soon, it would increase in Ethiopia from 43 to 630 cubic meter per capita storage volume. It is still too low for water security, for people, for energy, for crops, etc. And what is the reason why we are so slow to construct reservoirs if they are so essential for human well-being? And this is a picture which shows that very much ill-guided Western environmental NGOs managed in the 1980s to convince the World Bank and then also bilateral donors to decrease funding for irrigation, decrease funding for reservoir building in developing countries. You see in these curves the decline which lasted more than 2 decades, and it's difficult to prove, but I dare to opine that this deceleration and ignorance of rural development and agricultural development in the least developed or the poorest countries in the world triggered very much the unregulated urbanization and the migration trajectories which we are observing today. Let me come to an example. This is the southern part of the African continent, and if we now start at the global scale and try to characterize what are the problems we are facing there, I'm using a very, very simple indicator. In the world, the renewable annual surface and groundwater flow is 40,000 cubic kilometers per year, and this is generated on 150 million square kilometer landmass. If those are each 100%, the indicator of the world is 1. So the water to land relation for the whole world is 1. If we go to the African continent, then it is only 3,991 cubic kilometers per year runoff from 20% of the world land surface. So the value for the entire African continent is 0.5. Halving, so it is already a dry continent, irrespective that in the center, the Congo River is the second water richest river of the world. And if we go to the southern African tip of the continent, there is only 274 cubic kilometers per water per year from 4.7 million square kilometers. The indicator value is again halved, 0.023. And if we look at the Republic of South Africa, the southernmost tip of Africa, this value is 0.17. So you see, as you move towards the southern part of Africa, you are touching the southern dry belt, and the water availability in this part of the world is only 17% of that of the global average. Now, in this area, for the time being, about 6.9% of the renewable annual water resources are withdrawn for human, agricultural, industrial, etc. water use. What can be done to improve it? How can South Africa look better? And the key of this is storage, because there are, of course, seasonal water surplus and a very long seasonal water scarcity. For example, the only river in the southern part of Africa which has considerable runoff is the Zambezi River, about 117 cubic kilometer flow rate, around half of the Danube. And there are two huge reservoirs already in operation, the Kariba and the Cahora Bassa dams, of which the joint storage capacity is about the annual flow rate. It looks excellent, but in this part of the world, you usually need 2 or 2.5 times more storage volume than the average flow rate in order to account for several years of drought, subsequent droughts. It is true that dams and reservoirs have immense environmental impact on the rivers, and some of the pictures Professor Józsa showed already indicates what are the consequences for environment, for ecosystems, for the biotope, as a river as a biotope. Yet, without additional and specially well-distributed and well-managed storage, we are unable to provide, as of now, for human needs, and it will be even worse if climate change takes a course as it is predicted, because there will be even more very water-rich and even longer water-poor periods. These are definitely almost irreversible changes, so we have to be very careful where we implement them, and they are also needed to have interbasin transfers, again an anathema to many environmentalists, but the other option is if the water not going to you, you are going to water, and it means, again, migration. In Southern Africa, there are wonderful plans for different international organizations how many million hectares should be irrigated. I made a so-called back-of-the-envelope thoughts and calculations and came to the conclusion that for the present irrigated 2 million hectares as of 2003, probably not all this area is irrigated, but 2 million hectares are equipped for irrigation in the area, could only be increased by about 900,000 hectares, so roughly 50% more can be irrigated, and then we reached those economically and ecologically allowed limits of the water resources. This is, of course, very little development because the area is already massively dependent on food imports to feed its population. So we need not only water resources development, but dryland agriculture, tolerant cultivars, and very sophisticated irrigation technologies. But as of today, you see that 20% of the arable land is irrigated in developing countries and providing 40% of the agricultural crops and 60% of the cereals. Irrigation is indispensable in these conditions, and in the future it will be even more. If we look into South African Republic of South Africa proper, we see that it will be hit, likely will be hit by the forecasts by climate change very enormously, having up to 6 degrees higher temperatures by the second half of the century. And irrespective that this country has almost 600 large reservoirs, it has also very elaborate interbasin transfer systems. It is still not enough to feed its population. In addition to that, South Africa is itself a target of African migration within the continent. Roughly 9 African migrants within the continent account for 1 African migrant who is appearing in Europe. So just the proportions are almost 10 times more people are migrating between African countries as transcontinentally. And as I showed, water-wise, the development potentials are very finite. Climate change, as I showed, would probably hit South Africa even more than generally the whole world. And Africa's population is rapidly growing, and as I showed, developed African countries, which is for the time being target of African migration. The problem is that at Cape Town, the continent ends, and then it's only high seas. We won't be able to adapt by becoming maritime mammals. So if any migratory waves comes from Africa, and it's likely to come exceeding the present one's ultima ratio, it will be northwards. Thank you very much.
Okay, thank you very much for your presentation, Professor. To give a big picture about the resources and about the population and all these global challenges that we might face, it seems that we need more information exchange and more cooperation, because these tasks individually are over that one group or one research team can tackle. So now we are moving to the fourth and final topic of our today's conference, that is food, or let's say biological processes and more humane consequences. So first, may I ask Professor Gabriela Pocsfalvi, Institute of Biosciences and Bioresources, National Research Council of Italy, to give her presentation, The Plant Nano World, Emerging Plant Extracellular Vesicles and Nanovesicles for Enhancing One Health System. The floor is yours.
Thank you very much, Professor Stündel, for the invitation, and also I would like to say many thanks to Professor Remennik. I started a new and most probably flourishing collaboration recently. Yes, now we are switching to the plant and maybe the human food, plant-based food field. And I'm excited to talk a little bit today about this new nanometer-sized worlds of plants. So just to give you a short outline of the presentation, so at the beginning I will talk about plant extracellular vesicles and plant-derived nanovesicles. Then I would like to show how we isolate and characterize these nanovesicles in our laboratory, and the example will be actually the topic what we started the collaboration, and so it's ginkgo biloba, and I would like to show how we set up cell cultures and how we isolate these nanovesicles from the nuts of ginkgo biloba. Then in the last part I would like to talk about how we can load them some valuable for humans, or also for sometimes for vegetables or animals, useful ingredients or molecules, and the example will be loading the nanovesicles from ginkgo biloba with astaxanthin. So yes, actually extracellular vesicles, or EVs, this is a relatively new but really exciting field in biomedical research. So this has been mainly studied so far in the mammalian field, and usually they are called exosome microvesicles or apoptotic bodies. These are nano and micrometer sized structures and enclosed in a biomembrane. So these particles are designed by the nature and has been optimized through evolution, and therefore they have a remarkable compatibility, biocompatibility, and also a very high structural stability. Actually, these extracellular vesicles are studied in the mammalian field because recently it has been discovered that EVs are a sort of communication system, so they can be passed from one cell to another, but they can go also to distant cells, and then they will deliver to their biological content and make a biological effect. So they are such a sort of tool of communication between the cells. So it's in Hungary actually EVs are very well studied, and really there are a few very famous research group. So it's a quite well developed field in the mammalian community, I mean research community who are studying mammalian cells. There is one drawback of this research, and this actually the relatively low amount of EVs which are produced by the cells, because it's especially when we are thinking about to use them in as a biotherapeutic solution, or we would like to load them with some valuable, and we are trying to use them as a biotechnological platform. So for that reason, there are initiatives that try to find the new sources, not only mammalian cells, but also plant-based cells or microalgae-based or milk or different other new sources for extracellular vesicles. In this field, plants are one of the most interesting resources, and actually it was not obvious since the beginning that plant can secrete extracellular vesicles. But I usually like to show this image that it's not really so true, because already in the 60s with cryotem, there were at least this Halperin and Jensen who were studying the ultrastructural changes during the growth and the embryogenesis of carrot cell cultures, and they have seen that these tiny particles which are released from the multivesicular body, they can cross not only the biomembrane but even the cell wall of the plant cells. So actually plant can seems that they can secrete extracellular vesicles. And more studies which have been done in the past years, evidence is growing that they have precious roles in the cell wall remodeling, but also unconventional protein secretion, also transport RNA, and more importantly, even also the defense against pathogens. So they are used as a plant using them to defend against the pathogens, and also in the symbiosis between plant and microbes. So if we are going towards this topic, you can see that we gain more and more information that actually they have a role, and we can use them maybe in future agriculture or animal and plant and human health. In this presentation, I really would like to distinguish about two things: the plant EVs and the plant NVs. The EVs, the extracellular vesicles. So by definition, they are released by the cells extracellularly. So in this case, we can use, for example, plant cell in vitro plant cell culture and the condition media supernatant to extract the vesicles, or we can study the apoplastic fluid and use a sort of technique, vacuum infiltration to get out these vesicles. But in one of our study, for example, we use the hydroponic culture, and we extracted the vesicles from the hydroponic solution, from the root exudate. So these are the so-called pure extracellular vesicles, and the other, which is, even if I say that they are not pure, but they are also very valuable because they are very interesting nanomaterials. These are the plant nanovesicles. And the main difference is between them that we can obtain using homogenization. So this technique, the homogenization, will surely break up the the cells. So a lot of organelles will get out, and then we make our isolation. And in this case, the mixture, what we usually end up, it's a very complex mixture. So it's containing extracellular vesicles, but mainly other small nanometer-sized vesicles. And once we have this complex solution, we will be never able to get, there is no method today to the pure extracellular vesicles because they are structurally very, very, very similar. So when I was thinking about this presentation, actually I was thinking on this One Health system, and yes, in our laboratory we isolate nanovesicles and extracellular vesicles. Then we do a very thorough characterization, and it includes the physical, morphological characterization of these vesicles, and it's extremely important because otherwise, so we really have to be sure what is the material we are working. This is based on proteomics, omics, different omics studies, but also we use a lot of morphological studies, cryothem, SEM, size distribution measurement, with really very different methods. But then we arrive after that to questions. So yes, we have the vesicles, we were able to purify them, but we would like to know how is their biological activity. And this is a second very important question, and this is where our collaboration will start with the group in Hungary, because we have to be sure that the extracellular vesicles are not toxic, and what sort of bioactivity they have, how they are uptaken by the cells, and how they are cellular distribution. So once we know it, then those which are active specifically, for example, for human, we can use for human health, but we can have extracellular vesicles from the plant world, which can be used back to the plant health. So it's getting to be interesting to find a solution from this nanoworld which will communicate and put together all the One Health system. So different solutions, what we can catch up, I think, from this nanoworld. So in our laboratory, we really use a lot of resources, plant resources. So we produce both pure EVs and nanovesicles. Basically, the edible plant-derived nanovesicles we have been produced from citrus species, coconut, feijoa, grapes, strawberry, tomato, mainly in Italy, yes, ginger, ginseng, and also ginkgo biloba. So I'm getting to this nice word of ginkgo biloba, which is one of the most distinctive tree in the world, and it's important for plant, but also for the plant evolution, but also for the human society. It's a Chinese plant, it's a Chinese symbol, and actually there are so many interesting facts about ginkgo biloba that I would need 2 hours to talk about this one. What thing is that they can be used for their edible nuts, also as a herbal medicine, religion, and also in art. Since now we are in Debrecen, I just wanted to point out that there are some nice trees, ginkgo trees, also Debrecen. So if you have a little time, just go around. I found this one, for example, in the Bemter, which is this one, but there is a new planted trees, street trees at Nyugati Kiskörút. So yes, in this part, I would like to show how we isolate the vesicles and how we characterize them. So when we isolated the extracellular vesicles from ginkgo biloba, we set up from the ginkgo nut, we have taken out the embryo and the endosperm, so we set up this callus culture, and then we grown the cells and then established the so-called cell suspension culture, and we studied this cell suspension suspension culture, and now we are keeping it for years. So they are a sort of renewable source for extracellular vesicles. And this would be a little bit long to explain how we isolate, but the main method what we use for them for their isolation is the differential ultracentrifugation technique. So we make a series of centrifugation and separate pellet and supernatant. And the final one is the ultracentrifugation, when we have in the pellet a so-called EV-enriched biomaterial, and that one we can further purify with density gradient ultracentrifugation or size exclusion chromatography. So this is the method which takes a little bit of time. There are several other new methods, but we still stick on with this one. And this slide shows that when we work with the plant resource like the nuts from Ginkgo biloba, the process is really pretty much similar. The main difference is the homogenization, because here we homogenize the plant material and then perform the same differential ultracentrifugation, gradient density ultracentrifugation, and SEC methods for the purification. The difference is mainly that we obtain much more from the plant material than from the single suspension cell culture. So when we come to the characterization of this bionanomaterial, actually one of them is the size distribution and the yield. What we found when we compared the nut-derived vesicles with the cell-derived vesicles, that the nut-derived vesicles, as I told it, we produce in a much higher amount. Their size and size distribution, which is in both cases heterogeneous, but let's say quite similar. However, the yield, the particle yield per milliliter of starting material, we found higher in the cell suspension culture because they are so-called purer. They have less interfering compounds. So to characterize their content, protein contents, we usually perform mass spectrometry-based proteomics, and this is just to have an idea what sort of protein constituents have. But a sort of material usually have several thousand different types of proteins, so I wouldn't go into this one, but we usually pick up the most important, and these are like heat shock proteins, ATPases, also in the seed, several storage proteins, what we identified. In this very nice cryothem image, on the left side you can see the CCM EVs, and on the right side the nut-derived EVs. And actually we found one main and important difference is that this EVs are surrounded with a double membrane structure, while the nut-derived vesicles are usually single membrane enclosed nanoparticles. And maybe in this one you can see what I'm saying a little bit better. When we go further with the characterization, we usually like to have something EV-like density, which is 1.11, and so we usually in our lab we determine the densities of the particles. We do SDS page analysis to see the protein profile, and these are those characteristics what I told that important to define and to be sure that we are working with the vesicles. This experiment was very interesting because it's a sort of time point experiment. We were excited to know whether when we have more cells, then we can isolate more vesicles. But basically, how we expected, it was not true, but because as you see it in orange, the extracellular vesicles, which are produced at the 2nd, 4th, and 6th days of the culture, actually were quite steady, while the protein profile profile of them were quite similar. We are also very interested in the active ingredient, and because now this material is coming from the ginkgo biloba. It's well known that ginkgo biloba has two principal active ingredients, and these are the terpene-trilactones and ginkgo flavon glycosides. And when we go to the terpene-trilactones, actually they are the ginkgolides and bilobides, and because there were no method which we could use in our lab for the MVs and the NVs. So it means a very small amount of and very high sensitivity detection. We set up GCMS-MS-based methods for the selected reaction monitoring of these substances. And I just would like to show you really the results, that it was also interesting for us that when we see the nano and micro vesicles from ginkgo, they are enriched in these active ingredients, these active secondary metabolites of the plants. So for their use or for the biological activity, it could be a sort of important starting point or important point to know. So since we are still working on this bioactivity, today I just would like to show this last few slides on how we can use them for molecular delivery. So this is when we know that they are not toxic, and preliminary experiments shows us that they are not toxic. So we can select one of our substance what we like or what we would like to put inside the vesicles, and we have selected astaxanthin and used different methods for the loading, the sonication and direct loading, and we set up some methods, chromatographic methods to determine the entrapment efficiency and the drug loading capacity. And here on the left, you can see the tubes, the ultracentrifuge tubes, and astaxanthin has this very nice color, intense red color, and you can see that the particles, the nanoparticles, were enriched in this region, and in fact we got quite good efficiency of loading with the nanoparticles in this fraction 4, which would be this. So just I would like to come to the conclusion. Then yes, I think the first time we have shown that EVs can be harvested from plant cell suspension cultures, so it could be a good source material for a reproducible production of these interesting nanomaterials. So when we compare the ginkgo EVs to the nut NVs, we have seen several similarities, but also differences, what is important to know and point out. And I believe that this material can be a good or promising vector for future molecular delivery. And I hope that this exciting and ongoing experiments and collaboration with the group of Professor Remennik will continue on the ginkgo vesicles. And thank you for your attention. And I would like just to give credits to my lab and the people who worked on this topic and the grants. Thank you very much.
Thank you for your presentation. When I'm listening to that deep fundamental research somewhere in the molecular biology, it makes me always understand how important fundamental research is. Because when you understand these biochemical processes, these nano processes, these are a bit of similar in the plants and animals and also humans, so that gives a importance for the one health or the one world concept, and also gives fuel to the fundamental research and also for innovation. So thank you very much. Ladies and gentlemen, we came to the last presentation today, and that is kind of wrap up the whole animal and human line. Judith Remenyik, University of Debrecen, will present the title Inherited Diseases and Inherited Health, How Transgenerational Epigenetics Can Change the Processes. The floor is yours.
Thank you very much. Dear Chairman, ladies and gentlemen, you heard from Laszlo. I would like to present you inherited diseases and inherited health. The new research result of the last decade, especially in field of nutrigenomics and transgenerational epigenetics, encourage you to find those superfoods that can modulate the functioning of our body at the genomic level. Many decades ago, special attention was already given to certain ethnic groups who did not suffer from diseases of civilization or even cancer. They had long, healthy life. Researchers began to examine the geographical features and the genetic background, but in the end they identified lifestyle, distance from the cases of civilization and nutrition as the case. Author Dan Buettner has been working to identify longevity hotspots around the world from then a decade. Buettner found 5 zones, areas he called the blue zones, but fortunately there are many more. The Icaria, Icanava, Oglistra region, Loma Linda, Nicola Peninsula, and other areas and ethnic groups, Hunza Valley, Bhutan, Okinawa, and Abkhazians in the Caucasus region. The blood pressure and digestive problems, high cholesterol, cardiovascular diseases, cancer, and diabetes unknown the people living here. They mainly follow vegetarian diet and eat little meat. What can secondary metabolites do to extend our lives? They have a pleiotropic effect. As they are able to increase the activity of SIME enzyme and have an inhibitory effect of other enzyme. They regulate signaling pathway and down or upregulate expression level. Human epidemiological studies and feeding experiments on animal models have provided significant evidence that during the critical period of development, imbalances and metabolic disorders occurring during maternal nutrition can permanently affect the health of the offspring and may even appear in the following generation. This, the FETAL program, has led to a new hypothesis called developmental origins of health and diseases. I think every ethnic groups have superfood. Our treasures is sour sherry. as a result of plant breeding and health research work, Hungary has unique varieties of sour sherry. In Hungary, geographic and climatic conditions are excellent for sherry production, which results outstanding nutrition value of sour sherry, especially content of anthocyanins. Anthocyanins content of Hungarian sour sherry is 200 milligram in 100 gram. Accumulation of their bioactive molecules in sour sherry is unique in the world. Hungarian sour sherry is used in other countries. A new type, functional food and food supplement were developed sour sherry matrix. The product is as same order as royal jelly, storable for more without preservatives and additives. The bioactive compounds are with higher concentration in our products. For example, melatonin, anthocyanins, make compounds anthocyanins, cyanidins, free of monoglycosid, and cyanidin, free of rutinosid, and malvidin of free and 5O diglycosides. And vitamins, antioxidant, and tannins, etc. The active compound is present in the own biological matrix and show the physical and chemical stability of the active compounds, effect of natural matrix that imitate the in vivo synthesis, and accumulation of the selected drug. The biological system provides other beneficial chemical substrates that minimize biotransformation by being better substrates, inhibitors of metabolizing enzyme. Development of product manufacturing technology is a core technology, including free Maillard step without using of chemicals. The secondary products formation is minimal. Our products is non-allergic. It doesn't not contain additives. The bioactive compounds retain their bioactive activity. In vivo biological utilization of our products in high due to the fact that they get into the organ with their own matrix and the metabolites affect additive. It is suitable for the future processing, vegetarian cocktail, food flavoring and coloring. Storable for more years without preservatives and additives. It was found, the effect of anthocyanin was tested. I was found that hyperglycemic induced oxidative stress significantly suppressed by anthocyanin-rich sour sherry extract. The gene expression of the tested pro-inflammatory cytokines increased either hyperglycemic condition, but significantly reduced by sour sherry extract. Also increased endothelin-1 and endothelin convertase enzyme 1 expression due to hyperglycemic was reduced by anthocyanin extract. and nitro-oxidase synthase enzyme expression level was increased by the extract. Increased permeability of the epithelial and endothelial cells layer results in the outset of pathogenic mechanism in both cells type, cell-cell connection play a regulatory role in altering membrane permeability. As a result of inflammatory induction by TNF-alpha, redistribution of proteins was observed in KCO2 cell culture, which was reduced by anthocyanin treatment. In HUVEC cell culture, the decrease in protein and microRNA expression were more during that inflammatory induction with the compensate by anthocyanin. I'm sorry. Anthocyanin significantly elevate the levels of pro-inflammatory adiponectins such as leptin and MCP1, interleukin 6 and resistin, while decreasing the production of anti-inflammatory adiponectin and interleukin 10 distribution to the development of tissue inflammation, insulin resistance, and other symptoms of metabolic syndrome. You heard from Melinda about the effect of sorcery extract on the intestinal microbiota. We firstly prove that the sorcery extract matrix can inhibit human salivary amylase by binding to the active site of the human salivary amylase enzyme, therefore including sugars. The bond are therefore inhibiting acidification and ultimately carries. On the other hand, it inhibits Streptococcus mutant, the form of oral biofilm preventing oral health. We verify that sorcery matrix rich in anthocyanins make component cyanidins type compounds inhibited the human salivary amylase activity, catalyzed hydrolysis competitively. In our hypercholesteremia rabbit model experiment, we proved that homocysteine accumulates in the plasma as a result of damage of the transsulfuration pathway. Homocysteine competes with adenosyl methionine for binding to the DNMT, which can lead to passive loss of methylation in replication DNA. High plasma homocysteine level correlate with DNA homomethylation and atherosclerosis, non-alcoholic fatty liver disease, and metabolic syndrome. As a functional food, candidate tertiary extract can be used as a therapeutic epigenetic modulators in lifestyle diseases such as metabolic disorders, cardiovascular diseases, asthma, rheumatoid arthritis, non-alcoholic fatty liver disease, and metabolic syndrome. New results have helped to highlight that anthocyanins as anti-inflammatory bioactive food ingredient also regulate epigenome-modified enzyme activities, acetylation, methylation, phosphorylation, ribosylation, oxidation, ubiquination. Thank you for attention.
Thank you. It's always the most difficult task to be the last presenter, but succeeded. Thank you. Congratulations. Now we ended the presentations, and there's time for questions. I'm calling the audience to, if you want to ask questions to any of our presenters. Okay, then I have to be the last or the first, as always. That's the task of a moderator. I would like to ask from Professor Norbert Lukács about the plastics. We know that the nanoplastics are all around the aquatic environment, and especially in the marine environment. Do you have any data that is it relevant in the inland waters or inland arable areas? So how agriculture, like in Carpathian Basin, or aquaculture is influenced or threatened by that microplastics?
So thank you for these questions. It's very important, but we focus it to the microplastic and the micro particles in the now, and we focus it in our research in the compounds with the moving from the plastic material and the specific by to the sunshine influence and the other and the data about to the concentration and the amounts of the particles in the water in the ocean. I don't have. Sorry.
Yeah. Thank you. Okay, I made my first step. That's your turn now. Nothing. Okay. So I will not continue on that. Since we are proceeding with the program, it's about a bit of we are ahead of time. But if there is no any further questions, then I will close the second session. And as the program says, that we are at the closing ceremony and the poster award. That was in the call for this conference, that we are inviting those young researchers and PhD students to present their posters in this conference. And we decided to present one poster award. It was not an easy task, as always, because there were several posters from all aspects of this one health issue. So from microbiology to animal husbandry to food to environment and so on and so forth. But the organizing committee took the responsibility and the courage to declare one poster award. I do not want to detail that how even fields were and how difficult it was to choose that particular poster. But I will I will now call Professor János Tamás, the member of the organizing committee, to present this poster award.
Dear audience, on behalf of poster committee, I would like to thank and congratulate for all participants of the poster session. As you observe, the scientific level was really high, so not really selected the best posters. This only the subjective opinion of the committee. I would like to congratulate again for all participants, and I would like to offer the colleagues to publish the best results of the faculty scientific papers, ACTA, agriculture, and others. So, the best poster session winner name Elzafia Ali Hamid Muhammad and Károly Pál. I don't know here. Please. Please. This is the certificate for the research team. Congratulate again. And I give you this.
Thank you.
I wish all the best for you.
Okay. Thank you. So now we have some more tasks to do. The first one is that I would like to thank the organizing committee members and all my colleagues to help to organize this event. As you know, this is the first one in our faculty, but we would like to organize it in each 2 years, so it will be a biannual conference, typically and specifically for research done in our university, not just our faculty, but in our university, and to show the results done by our fellow researchers with our partner universities and research institutes. This would be rather scientific and fundamental oriented, because we believe that fundamental research is the important thing, even for agriculture, for the food industry, because you can build your activities there. And also I have to thank you the technical staff and also the media center, that they made the others who couldn't be here to follow this conference. So as I'm coming to the end of my talks, I had to give you some take-home messages. I took the courage and the opportunity. I did it myself, and there are some sentences and some ideas that I was experiencing when listening to the presenters today. One is that we have some huge extent of knowledge in biochemical processes or molecular processes that we have to increase further. And that's because if we have better understanding of these biochemical processes, we became to understand the living organism, the activities or the processes in the living organisms better and better, regardless of it's plant or animal or even human. And that activity should be the focus, because by the instruments or the technology available, we can do it better and better in the next future. So we possess the technology, machinery, equipment, also human brain, human resources, to do that, to find out these secrets, what is behind these processes. The second one is that the fundamental research has to have a focus, because we can build, as I said before, our practice-oriented or industrial research on that, and also the innovation. And then it's very important to do it in a sequence, in a line of research, including funding. Sometimes we forget to fund fundamental research or just give minor funding, and we say that the industrial one, the innovation, should take more from the national and international budget. Now, I hope that this conference is also drawing attention to the significance of fundamental research. The third is that now, as we heard that how diverse is the research done, even in our partnership about life sciences, it seems that it's not just only one health, but only one knowledge that exists. So let me draw the parallel between one health and one knowledge. If we exchange the knowledge, if we exchange our scientific results, living in an area like Carpathian Basin or the Tisa watershed, and in Australia, of course, so it helps us researchers here, or may help you researchers there, to find out the secrets behind these processes. So we have to add up together our results. If we publish it, it's open, but we have to draw the attention, and that's why it's good to meet, like in a conferences like this one, to draw attention to my or your experiments, your results, and help to understand these complex processes, and also draw attention to ones which are quite new even for the science. And the fourth and last take-home message I would like to give you is that bad decisions are originating from lack of knowledge or reluctancy in data collection and evaluation. We know that some politicians or decision makers are they do not hesitate to make decisions, and that has very significant consequences. Our task, scientists and education in higher education, is to draw the attention of the politicians, open their eyes, and say that do not make a decision suddenly or without any deep understanding of the problem behind. What I say is the decision maker has to have enough time to get the information to base the decision on. So, and it's our task then to do that, to translate that scientific information into sentences that they can understand, and make the science as a uniform language, and try to help them to make our lives easier and better. So I think that's it, what I wanted to say. I'm asking my fellow organizers, do they want to add something to it? If not, then I thank you for your presence, for your participation. And there is only one thing left, the conference lunch. It's in the university canteen, about 5 minutes walk from here. So if you start, you can take your time. I'm asking those colleagues of mine that they know where it is to help you to find that way. It will be a self-service thing, so you can find tables there. You can make groups if you want, and you can sit down and enjoy the meal. There will be several soups and several main dishes and some desserts, and you deserved it, so please enjoy your meal. Thank you for coming, and have a safe trip back home. Thank you for your attention.